الطاقم لها. التحديات أحيانا تكون في نقص معلومات أو في ضيق وقت. في حال كورونا مثلا كان كل أسبوعين عندنا اجتماع لدراسة الوضع الراهن، لكن الوضع على السفينة بحكم أنظمة التهوية فيها، فوجود أي شخص عنده كورونا راح يوقف المشروع. كل الجهات يتعاملون ويتبنون القضية كأنها قضيتهم. حرس الحدود، الهيئة العامة للموانئ، الجمارك وغيرها من الجهات اللي هم معك يركضون يسعون. غالبا ما كان سفينة في ميناء والسفينة الثانية في ميناء ثاني، فالعملية اللوجستية والربط والتواصل مع كل الأشخاص والأفراد، كيف نضمن أن السفينة موجودة في الميناء في الوقت المحدد بالتاريخ المحدد، وصول التموين، وصول الوقود، وصول طاقم العمل، نزول طاقم العمل، في تبديل بين الباحثين وبين طاقم العمل السفينة أيضا. شعور بفخر وباعتزاز، والأهم من ذلك أنه جميع الأشخاص موجودين على متن السفينة بصحة وبسلامة، جميع الأدوات، جميع الممكنات، كل شيء موجود ومتاح لهم أنهم ينجزون هذا. ومن ثاني يوم بدأت تطلع الاستكشافات سبحان الله.
ومع هذا الفيلم القصير نبدأ أول جلسة حوارية لهذا المنتدى بعنوان اللوجستيات والتحديات لمشروع عقد استكشاف البحر الأحمر. ومع هذا أود دعوة زميلي الأستاذ مصطفى فلمبان، باحث بيئي بحري، لتقديم هذه الجلسة وإدارتها.
Ladies and gentlemen, it's my pleasure to welcome you all to this panel discussion on the logistic and capabilities of the Red Sea Decade exhibition, Lessons Learned. This exhibition, which aimed to explore the biodiversity and habitats of the Red Sea, was a remarkable endeavor that brought together experts from various fields to unravel the mysteries of this unique marine ecosystem. Today, we have the privilege of having esteemed panelists who played pivotal roles in this exhibition. We are honored to have with us Dr. Vincent Prepon, co-CEO and Chief Science Officer of the research vessel Ocean X, who led the exhibition and oversaw its logistics and scientific objectives. Engineer Omar Abnayan, head of the Innovation Department in the National Center for Wildlife, who led the Red Sea Decade Exhibition project. Additionally, we have Dr. Faisal Al-Saag, Vice Dean and Head of Hydrographic Department in King Abdulaziz University, who contributed to the success of RSDE. And finally, Rear Admiral Commander Naji Al-Jihani from the General Directorate of Border Guard, who ensured the safety and security of the exhibition team throughout their journey. There is a devices that translate the discussion while going on, so please use them if you need them. Now, without further ado, let us delve into the heart of this discussion. I would like to start by addressing a series of questions to our esteemed panelists, and I will start with the first question addressed to Engineer Omar Al-Bunayan. Engineer Omar, the Red Sea Decade Expedition, a bold and large undertaking to explore the Red Sea along the Saudi coast. What was needed to make it happen, and when did this all start?
Thank you very much. That is a very interesting and very important question, actually. I would like to take you back to the 23rd of December, when the CEO of the National Center for Wildlife, Dr. Mohammed Qurban, looked over his shoulder and told me, now it starts the planning, now starts the mission, and now you need to do your work. And from that day on, we had everything in place related to scientific plans and also as logistic plans, getting all the permits needed, getting all the teams, all the visas, everything from everyone aboard. And I think the most important thing was having the scientific plan put in place, because it was the foundation of which we determined what kind of vessels were going to be used and equipment on these vessels, who will be on these vessels, the researchers, the scientists, the deck hands and everything else. And from that, we just kind of went down through the phases of getting all the permits necessary. First of all, the permit to have an exploration done, which is done by the General Authority for Geospatial Information. And then from that, we started going down the list of who are the different entities that we need to contact, what are the permits that we need to get done, who needs visas to come into the country and to be on this ship, what kind of logistics things need to be planned out, getting fuel ready for the ships, getting all the food and all the supplies and equipment for the safety, for the exploration. Everything had to be at the right time, at the right place, in order for this to happen. So what was needed was a lot of logistics and a lot of planning and a lot of cooperation, collaboration between all the entities, which is one of the major keys to the success of this Red Sea Decade expedition. I have Professor Vincent. He knows all about the last days before the expedition and the planning. We needed to get helicopter permits to use the helicopter on board the ship. We needed to get the permission to get all the drones. We needed permission to have all the equipment operational. And all of that came from the scientific plan. Once we had the scientific plan, we had our direction, we had our goal, we knew what we needed to do next. And to be honest, the main challenge that was surrounding the whole expedition was the fact that we were doing it during the pandemic of COVID-19. And I think that was one of the major challenges that we faced during this expedition, but then nonetheless, we managed to get everything done on time.
Thank you. That actually brings up a very good question mentioning the logistics. So logistics of people, supplies, assets, and much more. Can you tell us more about some of the challenges that were faced and the lesson learned from solving them?
Definitely, yeah. I would say we did face, first of all, the challenge of COVID. And since we are going to be embarking on research vessels, vessels in nature have a closed air system, which means if there's a virus on board, it will go everywhere. And everyone is in close encounters with everyone else. So the first mission for us was to make sure that we quarantined everyone that's going to go on board these ships and make sure that everyone understood the importance of this. At the same time, we had another challenge of logistics is finding the hotels at that time that can take 25 to 45 people at one time for quarantine, because we had ship crew, we had scientists and researchers and equipment operators are all coming in to get this task done. So finding that during the high season in Jeddah was a big challenge, let's put it lightly. But also after that was the unique design of the ship and its size also posed challenges with relations to fueling the ship up. The ship consumes or holds a lot of energy, a lot of fuel, and only one port at that time we were able to fuel that ship, and we needed to make sure that the fueling happened on time, because during the decade expedition we had changes of the personnel approximately every 10 days. And during that time we would stop at the port for only about 24 hours, sometimes a little bit more, sometimes a little bit less. And during that time we needed to make sure that all the food, all the supplies, all the fuel was on board at that time. Other than that, we always have the challenge of weather, but we cannot control that. That's out of our hands. But all in all, I think those were the two main challenges, and I don't think, and I hope we don't face another COVID in the future. Other than that, I think the lesson learned was great planning and great communication is the key to success.
What an interesting story is, Engineer Omar, and I can imagine the difficulties you went through. I have another question for you. What role did innovation play in the expedition, and how did it enhance the exploration and research process?
This whole project is innovation. This whole project is innovation. It's new equipment, new vessels, new techniques that led to the magnificent findings that we're about to present later on today. So I think innovation was the foundation of this project from the get-go.
Thank you, Engineer Omar.
You're most welcome.
Dr. Vincent, how did the research vessels' capabilities and technological advancements aid in the success of the expedition?
Thank you. I just would like to thank everybody for inviting me here. And I just also would just like to thank Dr. Orban from the very beginning for inviting us into this wonderful country to experience such a beautiful place and to have such an amazing expedition, and Dr. Carlos for helping plan and execute that expedition. As Omar said, this was very challenging for us. The vessel was very new. This was really the second expedition it had ever done. The first was done previously in the Red Sea, just up the coast. So we were just breaking the ship in, and to go to a very remote location was very scary for us and challenging, because we thought equipment could break and things like that. But it all performed amazingly, and the host country was amazing to us. There were a lot of things on the ship that I think really were perfect for this situation, because the ship was designed to do deep water and shallow water. And you're really blessed to have a country that has water that within a very short distance from the coastline is very deep. So we were able to drive in and out to the edge of the territorial waters and back into shore, and be able to cover very shallow water to very deep water in the Red Sea. So all the assets of the vessel, from the submarines and the ROVs, were in use, as well as shallow water mapping and things like that. We also used the helicopter extensively, which, as Omar mentioned, was challenging to get permits for. But I think we did something like 140 flights on the helicopter while we were there, with Nicholas, looking for megafauna, which will be presented later in the day. So this was a really all singing, all dancing, as we call it, expedition, because we had things in the air, we had things underwater, we were mapping day and night. So it was very, very busy. The team was exhausted by the end of it, which is a good sign, which means that the expedition went well.
Exactly.
Thank you, Doctor. Were there any unexpected challenges or obstacles encountered during the expedition, and how were they overcome?
So one that I can think of, which we should have known about, was the temperature in the Red Sea and how the temperature doesn't change as you go deep. And so we sort of understood this, but I don't think it really sunk into the entire team. So the problem is that when you have an ROV, when you have a submersible that has an acrylic hull, which you'll see throughout this exhibit, those acrylic hulls are rated for a certain depth, and that depth is dependent upon temperature and pressure. And everywhere else that we've ever been in the world, or those submersibles have ever been, at about 1,000 meters, the temperature is almost 4 degrees centigrade. Well, we were in water that was 18 to 22 degrees centigrade. And so we had two problems. One is that we had to reduce the operating depth, because at that temperature and depth, acrylic tends to flow. And flowing is not something you want to hear for your solid hulled submersible. We also had a lot of acrylic domes and spheres on cameras, and those also leaked. And that was only because of the heat and the temperature of the acrylic. It becomes soft with that amount of pressure at that depth. So we solved it all. We replaced a lot of the hoods with crystal and everything before we came back. But that was a challenge that we didn't expect. And we had a small ROV that we would put down. The plan was to put the small ROV down into one of these beautiful brine pools that you have. And that didn't last very long because the temperature in that brine pool was very high. It was one that I think Carlos had discovered years earlier. And the ROV immediately went into overload, and that was the end of that little expedition into the depths of the ROV. So I think the temperature and the salinity are quite a challenge when you work in the Red Sea.
That's very interesting, Doctor. Let's have another point of view of the challenges faced. Dr. Faisal, what were the main logistical challenges faced during the Red Sea Decade expedition, and how were they overcome?
First of all, I would like to thank Dr. Mohammed, CEO of NNCW, to hosting me in this event. And also, we are happy as King Abdulaziz University was a participant in this expedition. Regarding about your question, as you know, when you are working in the sea, especially, you have a lot of challenge, like permits, weather condition, and so on. But in Red Sea Decade expedition, that was different kind of challenge, which was COVID-19. This is make us be honest. That's too hard. It's a big challenge. How to prevent this virus to be entering in the vessel? How to keep the crew protected of this infection? But end of that, we try to keep the project running. Alhamdulillah, end of that, it was some delays, be honest. We have some positive cases. We overcame these problems, but end of that, alhamdulillah, project was running.
Thank you very much, Doctor. So the tasks of border guard, including protection and security of the kingdom border, and keep watching the Saudi maritime zones. Commander Naji, how to keep safety and security of the local and international agencies carrying out exploration and research activities inside and outside of the Saudi maritime zones? And what are the challenges faced border guard in this project or in this respect?
Okay, thank you. First, I would like to thank the National Center Wildlife to invite the border guard to attend in this Decade Symposium. The border guard tasks be as important for the security and the safety for all those ships patrolling for any missions in the Saudi water zones. And as we know, the Saudi border guard already, they established since 15 years from now 2 international centers for the rescue and for maritime-wise. In this time, just I want to clarify some tasks belong the border guard. All the patrolling vessels, even they use for the pleasure boats or for the expeditions, I hope that to be contact with these centers in Jeddah and the other similar one in Dammam, because we established these centers just for search and rescue and for the safety and for the security-wise. And also, all these centers equipped with all the transponder for the search and rescue. And in this side, just advice for all the patrolling vessels to contact directly with these centers and give them the route and the plan for all these missions, because in case of emergency, that will be reply for any transponder from the EPIRB or other satellite devices we can receive through this one. And also now we're already monitoring in these centers with the AIS Live and IIS devices that we receive all the plan and the route for all this. And in case of emergency, just we receive any distress alert to be transponded from any, transmit from any transponder. It's be on board these ships. And now the border guard also, through ZAUL application, that's the easiest way now to fill all the data when you want to plan for any missions, even for the expedition or the others. That's mean now the border guard with all this unique application, that's me help and support all the entities they need to go anywhere for any missions.
Thank you very much, Commander. Going back to Dr. Faisal, in your opinion, Doctor, what is the next step and what do you think the future looks like for national research vessel here in Saudi?
I think the next step is to keep going, working together in any kind of marine studies. I think the most important is how to collaborate between us. What that means us between government, semi-government companies and universities. And we have a good experience, to be honest, with working with King Abdulaziz University and NCW, working in the marine studies at Red Sea last year and this year. Regarding about the national vessel centers in Saudi, I think, as you know, we have a good distribution of vessel centers, or vessels. That's one, as you know, Al-Azizia and Thawal in Red Sea. And in Arabian Gulf, we have Taqniya and Sultan. And in addition, I'm not sure, maybe Dr. Muhammad allow me to announce this. We have also a new fleet from NCW, will be in Red Sea and Arabian Gulf. I think with these vessels, with these boats, can be doing any distinguished studies in both Red Sea and Arabian Gulf.
Doctor, in terms of future expeditions and research initiatives, what are the key recommendations or lessons learned from the Red Sea Decade expedition that can guide similar endeavors and in exploring other marine ecosystems?
Yeah, as I mentioned before, we need a cooperation between us. I think with the vessels that we have, and we have a scientist, we are interested working with the international centers and experts. I think with these tools, I think it will help us to cover all Saudi water.
Thank you very much, Dr. Faisal. Dr. Vincent, how did the Red Sea Decade expedition contribute to raising awareness about the importance of marine conservation and the need for sustainable practices in the Red Sea region?
One of the key deliverables that we felt we needed to produce in this decade expedition is a series of media projects, which were films, social media posts, things like that, that would raise awareness, not just locally, but also internationally, about the beauty and the sort of pristine nature and unique nature and preserved nature, actually, of the Red Sea, especially on the Saudi coast. We were first invited, as it were, to come here by His Royal Highness, who expressed to us a desire that as future development happens within the Saudi Red Sea, that all possible precautions be put in place to ensure that you can maintain this pristine environment. So I think from that perspective, that to us was sort of the greatest sign that this beautiful thing that you'll see over the next 2 days and what you'll see in videos and in films will be retained, and that all the secrets that are locked in these incredibly warm, incredibly salty, but still very robust and diverse environment can be maintained as development happens and as the future goes on.
Thank you very much. And by that, I thank our panelists who gave us valuable lessons from the Red Sea Decade exhibition, and I thank all those who attended this panel discussion. Thank you very much.
Thank you, Mr. Mustafa Flimban. And with that, we have finished our first panel discussion. We will have a coffee and refreshment break right now, and hopefully we will rejoin at 12:15 p.m. for the inauguration of this symposium.
call the Red Sea the baby ocean. It contains all the features of any ocean. Red Sea is very unique that you need to understand it well. And this is what we did in this cruise. And this is what we call the decade expedition for the Red Sea.
What we're doing in the Red Sea Decade Expedition is
We call the Red Sea the baby ocean. It contains all the features of any ocean. Red Sea is very unique that you need to understand it well. And this is what we did in this cruise. And this is what we call the decade expedition for the Red Sea.
what we're doing in the Red Sea Decade Expedition is bold, and it has not been done before.
The Red Sea Decade Expedition embarked on an unprecedented journey into one of the expedition embarked on an unprecedented journey into one of the world's most captivating marine ecosystems. This extraordinary expedition delved into the unparalleled beauty and scientific significance of the Red Sea.
We call the Red Sea the baby ocean. It contains all the features of any ocean. Red Sea is very unique that you need to understand it well. And this is what we did in this cruise. And this is what we call the decade expedition for the Red Sea.
What we're doing in the Red Sea Decade Expedition is bold, and it has not been done before. I'm not aware.
طبعا أهمية
We call the Red Sea the baby ocean. It contains all the feature of any ocean. Red Sea
where they are going to be.
Expedition for the Red Sea. And this is what we did in this cruise. And this is what we call the decade expedition for the Red Sea.
What we're doing in the Red Sea, they can that any nation.
We call the Red Sea the baby ocean. It contains all the feature of any ocean. Red Sea is very unique that you need to understand it well. And this is what we did in this cruise. And this is what we call the decade expedition for the Red Sea.
What we're doing in the Red Sea Decade Expedition is bold, and it has not been done before.
It was amazing because we caught a lot of sharks, at least 4 different species, and one of them is the scalloped hammerhead, and it's a critically endangered shark.
Our research that we are doing now can be in the biology classes tomorrow in our school.
The coral reef is well developed all along the Red Sea, which is a very, how can I say, it's a very weird basin because it's an enclosed basin and it has the highest salinity and all
planning for be able to do that in
We call the Red Sea the baby ocean. It contains all the feature of any ocean. Red Sea is very unique that you need to understand it well. And this is what we did in this cruise. And this is what we call the decade expedition for the Red Sea.
What we're doing in the Red Sea Decade Expedition is bold, and it has not been done before. I'm not aware that any nation has tried to produce an inventory of their marine biodiversity through an approach based on environmental DNA. To be able to do that in a research vessel is even more unprecedented.
One of the many advantages of eDNA is that we do not rely on the organisms actually being present at that very moment. Since we're observing wildlife, we cannot predict where they are going to be and how many are there going to be.
After 8 months of planning for the Red Sea Decade Expedition,
this what we call the decade expedition for the Red Sea.
What we're
We call the Red Sea the baby ocean. It contains all the feature of any ocean. Red Sea is very unique that you need to understand it well. And this is what we did in this cruise. And this is what we call the decade expedition for the Red Sea.
What we're doing in the Red Sea Decade Expedition is bold, and it has not been done before. I'm not aware that any nation has tried to produce an inventory of their marine biodiversity through an approach based on environmental DNA.
We call the Red Sea the baby ocean. It contains all the feature of any ocean. Red Sea is very unique that you need to understand it well. And this is what we did in this cruise. And this was we call the Red Sea the baby ocean. It contains all the feature of any ocean. Red Sea is very unique that you need to understand it well. And this is what we did in this cruise. And this is what we call the decade expedition for the Red Sea.
What we're doing in the Red Sea Decade Expedition is bold, and it has not been done.
Ocean. It contains all the feature of any ocean. Red Sea is very unique that you need to understand it well. And this is what we did in this cruise. And this is what we call the decade expedition for the Red Sea.
What we're doing in the Red Sea Decade Expedition.
We call the Red Sea the baby ocean. It contains all the feature of any ocean. Red Sea is very unique that you need to understand it well. And this is what we did in this cruise. And this is what we call the decade expedition for the Red Sea.
What we're doing in the Red Sea Decade Expedition is bold, and it has not been done before. I'm not aware that
We call the Red Sea the baby ocean. It contains all the feature of any ocean. Red Sea is very unique that you need to understand it well. And this is what we did in this cruise. And this is what we call the decade expedition for the Red Sea. We call the Red Sea the baby ocean. It contains all the feature of any ocean. Red Sea is very unique that you need to understand it well. And this is what we did in this cruise. And this is what we call the Red Sea the baby ocean. It contains all the feature of any ocean. Red Sea is very unique that you need to understand it well. And this is what we did in this cruise. And this is what we call the decade expedition for the Red Sea.
What we're doing in the Red Sea Decade Expedition is bold, and it has not been done before. I'm not aware that any nation has
The Red Sea Decade Expedition embarked on Red Sea Decade Expedition embarked on an unprecedented journey into one of the world's most captivating marine ecosystems. This extraordinary expedition delved into the unparalleled beauty and scientific significance of the Red Sea, revealing its mysteries and marvels. Led by the National Center for Wildlife in collaboration with King Abdullah Science and Technology and numerous partners, the Red Sea Decade Expedition brought together 126 experts, utilizing OceanX's Ocean Explorer and King Abdulaziz University's Al-Azizi vessels, equipped with state-of-the-art technologies. The team geared up for a 19-week voyage, aiming to study the marine biodiversity from start to finish, from the smallest organisms to marine mammals, and to establish a baseline for sustainable management.
We have the best 2% coral reef in the world.
The expedition's profound exploration uncovered remarkable findings. DNA sequencing confirmed the presence of great white sharks, previously unseen due to their rarity. Active millenary-old chimneys, supporting unique fauna, and a remarkable set of over 20 blue holes and sunken lagoons were discovered, offering promise for conservation and ecotourism. The team's groundbreaking work unveiled a thriving population of mesopelagic fish with a remarkable density and tolerance of very low oxygen conditions, providing a foundation for the food web. Breeding bride's whales were detected for the first time, and seafloor animals in the Red Sea were observed preying on mesopelagic fish during their migrations, a phenomenon never. The Red Sea Decade Expedition delivered a unique inventory of biodiversity, from bacterial to whales, providing a baseline of the health status of key habitats and species of conservation interest, serving as a resource to plan and manage marine protected areas.
The deepest scattering layer is not only a thick layer of fish, but now we understand that many animals in the ocean rely on this layer as food.
The Red Sea Decade Expedition not only delivered groundbreaking scientific discoveries, but also established the Red Sea as a national heritage with potential applications in various sectors. This ambitious project has redefined our understanding of the Red Sea.
Expeditions like this come up once in a lifetime.
Many expeditions happen there in the sea, many studies, but not like this time.
Shaping the future of marine conservation and scientific exploration in this unique marine ecosystem.
بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. معالي وزير البيئة والمياه والزراعة، رئيس مجلس إدارة المركز الوطني لتنمية الحياة الفطرية، معالي المهندس عبد الرحمن بن عبد المحسن الفضلي، أصحاب السمو، أصحاب المعالي والسعادة، الحضور الكريم، السلام عليكم ورحمة الله وبركاته. أنا عمر أبو نيان من المركز الوطني لتنمية الحياة الفطرية، مدير مشروع رحلة العقد لاستكشاف البحر الأحمر، ويشرفني هذا اليوم أن أقدم لكم ندوة رحلة العقد لاستكشاف البحر الأحمر. Your Highnesses, Your Excellencies, esteemed guests, and distinguished participants, it is my great honor to welcome you to the Red Sea Decade Expedition Symposium. I extend a warm welcome to the Honorable Minister of Environment, Water and Agriculture, Chairman of Board of Directors to the National Center of Wildlife, His Excellency, Engineer Abdurrahman bin Abdul Muhsin Al-Fadli. Now it is my pleasure to introduce Dr. Muhammad Ali Qurban, the CEO of the National Center for Wildlife, who will deliver the welcome remarks. Dr. Qurban has been an instrumental part of the Red Sea Decade Expedition, and his expertise and dedication have significantly contributed to the success of this groundbreaking mission.
معالي وزير البيئة والمياه والزراعة، رئيس مجلس إدارة المركز الوطني لتنمية الحياة الفطرية المهندس عبد الرحمن بن عبد المحسن الفضلي، أصحاب السمو، أصحاب المعالي والسعادة، الحضور الكريم، السلام عليكم ورحمة الله وبركاته. يطيب لي بداية أن أرفع أسمى آيات الشكر والامتنان إلى مقام سيدي خادم الحرمين الشريفين الملك سلمان بن عبد العزيز وولي عهده الأمين صاحب السمو الملكي الأمير محمد بن سلمان بن عبد العزيز رئيس مجلس الوزراء يحفظهم الله على عنايتهم الكبيرة واهتمامهم بكل تفاصيل التنمية في ظل التحولات الكبرى في بلادنا المتواكبة مع رؤية المملكة 2030. كما أشكر معالي وزير البيئة والمياه والزراعة رئيس مجلس إدارة المركز الوطني لتنمية الحياة الفطرية على جهوده البارزة ومتابعته المستمرة وحرصه على الارتقاء بالمنظومة البيئية وتحقيق التكامل المثمر في مخرجاتها كما يلبي احتياجات التنمية. وأرحب بكم جميعا ضيوفنا الأعزاء في هذه الندوة التي سنبحر من خلالها مع رحلة العقد الاستكشافية التي نظمها المركز الوطني لتنمية الحياة الفطرية بالتعاون مع الجامعات المملكة العريقة من خلال سفينتي الأبحاث العالمية أوشن إكس والوطنية العزيزي، وبمشاركة 126 باحثا وعالما لإجراء دراسة شاملة للمياه السعودية في البحر الأحمر الممتدة من منطقة عفيف جنوبا حتى خليج العقبة شمالا، والتي استثمرت أكثر من 19 أسبوعا. الحضور الكريم، لقد جاءت هذه الرحلة العلمية امتدادا لجهود المملكة التي حققت قفزات توعوية في مجال التنمية المستدامة التي تشمل حماية البيئة وتأهيل نظمها وتوازنها وإثراء التنوع الأحيائي فيها، في ظل حرصها على دعم الجهود العالمية التي تثري مسارات التنمية البيئية. إن تنظيم هذه الندوة التي ستقف على نتائج رحلة العقد الاستكشافية يندرج ضمن سلسلة الجهود التي يبذلها المركز الوطني لتنمية الحياة الفطرية بالتعاون مع شركائه لحماية النظم البيئية واستدامتها وازدهارها والحفاظ على التنوع الأحيائي ودفع عجلة النمو الاقتصادي وتحقيق جودة الحياة في ظل حرصنا على تطبيق أفضل التجارب العالمية التي تساهم في تشكيل المشهد البيئي المستدام بما يتسق مع الاستراتيجية الوطنية للبيئة ومستهدفات مبادرة السعودية الخضراء ورؤية المملكة 2030. لقد استهدفت الرحلة خوض غمار البحر الأحمر لدراسة خصائصه البيئية والتنوع البيولوجي الفريد فيه، إلى جانب وضع خرائط تفصيلية لتوزيع الموائل البحرية كالشعاب المرجانية والحشائش البحرية، وتقييم المخزون البيئي للكائنات الكبيرة كالثدييات البحرية والسلاحف وأسماك القرش، إضافة إلى توفير مواد إعلامية وتعليمية تساهم في رفع الوعي البيئي وتوحيد جهود حماية البيئة وتقديم الحلول النوعية لتحدياتها، وصولاً إلى تطبيق استراتيجية تعزيز الاستدامة البيئية وتحقيق التنمية الاقتصادية والاجتماعية والحفاظ على الموارد الطبيعية وتعظيم ميزاتها التنافسية مستخدمة أحدث الوسائل التكنولوجية للرصد والمراقبة التي ساهمت في توفير كم كبير من البيانات الدقيقة التي تمثل مصدراً غنياً للبحث والتحليل والاستنتاج، بما يساهم في فهم الأنظمة البيئية ودعم اتخاذ إجراءات تساهم في الحفاظ على الأنواع المهددة بالانقراض. وتأتي ندوتنا اليوم لاستعراض قصص النجاح الاستثنائية التي سجلتها هذه الرحلة التي سبرت أغوار البحر وكشفت أسراره وخباياها العجيبة وكنوزه المغمورة الفريدة، وخرجت بحزمة من الاكتشافات المبهرة الأحيائية والجيولوجية والبيولوجية، ولتصبح هذه الندوة منصة عالمية للتبادل المعرفي والعلمي المبني على نتائج هذه الرحلة الثرية من خلال محاضرات الخبراء المحليين والدوليين وجلسات النقاش الإثرائية. الحضور الكريم، بعد رحلة العقد نستطيع القول إننا نملك خط أساس صلب ننطلق منه لإدارة ثرواتنا البحرية مما تحتويه من نظم بيئية وتنوع إحيائي، إدارة مستدامة لازدهار حاضرنا وضمان مستقبلنا لأجيالنا، متطلعين إلى نجاح هذه الندوة والاستفادة منها ومن نتائج رحلة العقد لتوحيد الجهود المشتركة في حماية وتنمية بيئة البحر الأحمر. والسلام عليكم ورحمة الله وبركاته.
And now we will have the privilege of hearing from Professor Carlos Duarte, who will deliver the keynote speech. Professor Carlos Duarte is an Ibn Sina Distinguished Professor from KAUST. Professor Carlos' profound insights and expertise in marine science and conservation will undoubtedly provide invaluable perspectives on the significance of the Red Sea Decade Expedition and its implication for the future of marine conservation.
Your Excellency, Minister Al-Fatli, CEO of the National Center of Wildlife, Dr. Osama Fakia, Your Royal Highnesses, Excellencies. So it's for me an honor and a privilege to deliver this keynote summary of the results so far of the Red Sea Decade Expedition. But before I do that, I would like to broadly celebrate the rising role of the Kingdom of Saudi Arabia as a global leader in environment. Yesterday, we were celebrating the International Day of the Arabian Leopard. So for the first time, one of the big cats of the biosphere has an international day recognized by the UN. And that celebration was also a celebration of success and collaboration because it brought together the efforts of the National Center of Wildlife to rewild the lands and protected areas of the kingdom, also the efforts of the National Center for Vegetation Cover to restore the vegetation of the kingdom, and the efforts of the Royal Commission for Alula to release the bred Arabian leopards into the lands of Alula, back to your kingdom and your lands. But it also was accompanied by a global campaign led by Her Royal Highness Princess Rima bin Bandar Al Saud, called Catch Fear, that invited citizens around the world to walk and unite themselves in to celebrate the conservation of the Arabian leopard and other big cats in the world. Today we move from land and leopards to the ocean, but again, it is a celebration of collaboration and a celebration of the rising role of the Kingdom of Saudi Arabia as a global leader in conservation. So before I deliver my keynote, I'd like to invite engineer Omar Abu Nayan and also Dr. Shannon Klein from KAUST to provide a very quick summary of the key discoveries of the expedition so far.
DNA sequencing confirms the presence of great white sharks in the Red Sea, which are so rare that they have never been seen and could only be detected through advanced environmental molecular technologies.
Active millenary-old chimneys represent microbial skyscrapers formed by microbial growth-releasing fluids. They were discovered north of the Farallon Banks and support unique fauna.
A remarkable discovery of numerous blue holes and sunken lagoons, of which 4 were surveyed, harboring beautiful coral walls, unique ecosystems. They have been discovered north of Jazan, which offer promising for conservation and ecotourism.
Sharks extend deeper in the warm waters of the deep Red Sea, with a minimum temperature of 21 degrees Celsius.
The Red Sea harbors a thriving population of mesopelagic fish with a remarkable density and tolerance of very low oxygen conditions, providing a strong foundation for the food web.
Breeding Birdie's whales were detected in the Red Sea for the first time, between Al Comforter and Al Leith.
Seafloor animals in the Red Sea predate on mesopelagic fish during their vertical migration, which was never reported before anywhere in the world.
Plankton in the deep, dark Red Sea are remarkably still photosynthetically active.
We detected unique dolphin behavior, where dolphins and their calves were observed within shelter of blue holes. At present, it is unknown if this is a purposeful way to avoid predation.
DNA sequencing coupled with age assessments of Red Sea floor sediments were used to reconstruct changes in biodiversity of Red Sea species since the year 1800 to date, providing a unique assessment of changes over time.
Plankton communities changed from diatom-dominated in the southern of the Red Sea to dinoflagellate-dominated in the northern Red Sea, shaping the food web of the open waters of the Red Sea.
The Red Sea delivered the largest ever collection of Red Sea crust, informing on volcanic processes shaping the Red Sea, but also supporting unique biodiversity.
Deep water corals, present down to over 1,000 meters in depth in the Red Sea, are the toughest coral organisms ever reported, uniquely able to survive extended periods without oxygen at temperatures up to 33 degrees Celsius.
A first catalogue of genes of Red Sea microbes that informs the genetic resources of the Red Sea as a national heritage with applications in cosmetic, pharma, food industry, energy, and environmental sectors. And with this list of impressive discoveries, I would like to invite distinguished Professor Carlos Duarte back to the stage for an in-depth overview of all of these discoveries. Thank you.
Thank you very much, Omar and Shannon. So now I will unpack a bit those discoveries, but I will provide just a summary of what we have done today, because we have 2 days of symposium and my colleagues will provide details, so I would not like to steal the thunder from the discoveries, and I will just provide a summary of what we found. And let me first indicate what was the overall goal of the Red Sea Decade expedition, which was to provide a baseline of biodiversity end-to-end. So we are providing inventories of biodiversity all the way from bacteria to whales in the Saudi waters of the Red Sea, with the goal of informing conservation efforts, but also to unlock the potential of marine biological resources to contribute to a thriving economy in the Kingdom of Saudi Arabia. In fact, we are providing the underpinnings for three major international commitments of the Kingdom of Saudi Arabia. One of them is the commitments under the Convention on Biological Diversity, and the information that the Red Sea Decade expedition delivers will be fundamental to establish 30% of the Red Sea waters for conservation under the commitments acquired with the Kunming-Montreal Global Biodiversity Framework. It will also identify degraded habitats that need to be restored under the commitment to restore 30% of degraded habitats by 2030, and it will also inform the obligations and the opportunities of the Kingdom of Saudi Arabia under the CITES Convention that regulates the trade of endangered species, and for which Dr. Mohammed Kurban is the point of contact in the Kingdom, and also for the Nagoya Convention that regulates the ownership of genetic resources of the waters of the Red Sea, which had been poorly explored to date, and this expedition then provides a catalog of genes and molecules of interest to be able to support a number of industries. So at the time the Red Sea Decade expedition was being planned, the National Center of Wildlife was, if I'm not mistaken, only 2 years old. So it was a very new organization that was still establishing itself, rooted in the heritage of the Saudi Wildlife Commission, but it was the first time that an expedition of this scale in the Red Sea had been planned ever in history. So not in recent years, not in the Kingdom of Saudi Arabia, but ever in history. And that was even planned and organized during a pandemic, the pandemia, so that added a challenge to the organization, but it was also a test of the power of collaboration. So the Red Sea Decade expedition was a collaborative program under the leadership of the National Center of Wildlife that brought together KAUST, KFUPM, KAU, the Red Sea Global, NEOM, and also international organizations like OceanX that not only provided the vessel, but also scientific support and extremely skilled crews and technical support for the expedition, and also intellectual contributions to render the expedition successful, and also NHK, which is the national Japanese TV, which has already premiered in Japanese documentary from the Red Sea Decade expedition, which is now being released in English with the voiceover of Sir David Attenborough, and is going to compete in international films. So there's also a strong focus on communication that we are seeing today, and I believe that we are joined by international media here in the symposium that will communicate also the wonders of the Red Sea to global audiences. So the way the expedition was designed was to cover the whole of the economic exclusive zone of the Red Sea, of Saudi Arabia in the Red Sea, from south to north, along 4 major segments that were each surveyed from the deepest water in the center of the Red Sea to the shallow coastal environments near shore. So all the way from mangroves to the deep sea. And we worked with 3 vessels. So OceanX, which was the vessel that was in charge because of their advanced capabilities for deep sea research to survey the waters deeper than 150 meters. Then a KAU Al-Assisi vessel that supported the work in shallow coastal environments, and even 1/3 vessel that is carried on board, OceanX, called Metal Shark, that was responsible for surveying the very challenging blue holes and other areas that are extremely difficult to access by conventional research vessels. And along all of these transects from shore to the deep sea, then we collected samples of plankton, of sediments, and also surveyed biological communities and habitats to provide that inventory of biodiversity end-to-end from the south to the north of the Red Sea. This is a hugely important resource for the kingdom to be able to have an assessment of your resources that will be propelling the blue economy in the future. And we also see a way to link the work we did with the newly established and announced strategy for biotechnology in the kingdom, because in the 21st century, marine microorganisms carry significant potential to contribute to industrial applications that will resolve problems in energy, agriculture, environment, food sectors. And the global value of this industry is about $6 billion per year, yet with very little contribution from the Kingdom of Saudi Arabia, but that market is doubling every 4 years. So we hope to align those two strategies to render the Kingdom of Saudi Arabia a globally relevant actor in the blue economy through marine biotechnology. So this is a summary of all the stations, all the locations where we collected samples from the different vessels. And basically, we were working offshore with OceanX and working on submersibles, but also ROVs and deploying other instruments, while we surveyed the shallow coastal waters of the Red Sea with a research vessel Al-Assisi from KAU. In the shallow waters, we conducted transects by scuba diving to be able to take images of shallow water habitats, including mangroves, seagrass meadows, algal beds, and coral reefs. And that led to an assessment of the health status of these ecosystems. So we are now analyzing the data, but we already made assessments of the health status of seagrass meadows in the Red Sea, and also the coral cover in the Red Sea from south to north. We observed that unfortunately the coral reefs in the southern Red Sea have been degraded due mostly to a global bleaching event that impacted the southern Red Sea in 2015. So the live coral cover is relatively small, but as we move north, the cover of living corals increase, and also that's biodiversity. But overall, the assessment showed no sign of disease to the corals in the Red Sea, and also very limited predation by a pest that often creates problems in Australia called the crown of thorns starfish. And then at the time there were very few bleached colonies of corals, although unfortunately in 2023, in around September, August to October, we have also suffered a bleaching event affecting much of the Red Sea. So in addition to assessing biodiversity through visual methods, we also have used advanced acoustics to be able to capture the soundscapes of these ecosystems that contain a fingerprint of fauna that is very difficult to see because they live within the crevices of coral reefs, but the sounds can be detected by hydrophones, and they can be analyzed using AI to be able to create compound indices of biodiversity in Red Sea coral reefs and other environments, which actually are the first soundscapes of coral reefs in the Red Sea that are being recorded. As indicated earlier, we were privileged to be able to discover and explore a number of blue holes and also sunken lagoons north of Jasan in a labyrinth of coral reefs that have very difficult topography and are very challenging to navigate. So here we benefited strongly from the mapping capabilities of OceanX and also the metal shark vessel that goes on board OceanX that has a very shallow draft, so it can enter very shallow environments. And we discovered these amazing blue holes that have unique ecosystems inside them, also have amazing vertical walls of corals outside them, and that should be in the future in the bucket list of all scuba divers around the world. So it's really a privilege to be able to scuba dive and observe these beautiful blue holes in the Red Sea. We also studied the calcifying algae that grow in coral reefs that together with the corals themselves form the main habitats of coral reefs. And there have been very little work on calcifying seaweeds in the Red Sea that are very important habitat forming organisms. And the Red Sea Decade Expedition has provided the largest collection, probably not only in the Red Sea, but also anywhere in the ocean, of calcifying algae that are going to deliver a catalog of biodiversity of this important habitat-forming species. Our efforts also focus on exploring the deep water corals of the Red Sea that are indicated earlier, have been discovered to grow down to 1,400 meters depth and form very unique ecosystems that also support habitats and a lot of different species in the deep waters of the Red Sea, forming habitats around 200, 300, 400 meter deep. These deep sea coral communities are very unique because in the global ocean they grow in very cold waters, around 4 to 2 degrees centigrade, but in the Red Sea the water is very warm all the way to the deepest part of the Red Sea, with the minimum temperature being 21 degrees. So these corals that grow elsewhere at about 2-4 degrees are growing at 21 degrees in the Red Sea, and it's not surprising, therefore, that we're finding a lot of unique species, not only species, but also genus and high-level taxonomies, like families of organisms that are unique to the Red Sea. We keep discovering new species and new genus and families, and we will greatly increase the catalog of species known and also unique to the Red Sea through the Red Sea Decade Expedition. We're also doing work to understand the growth form of these species, to be able to not only model them, but eventually, where needed, be able to restore these habitats where they have been damaged, although we found deep sea corals to be generally in good health status, because they have not been impacted by impacts that affect shallow water corals, like climate change and other impacts. So we characterize the environmental conditions of the Red Sea. In the upper layers of the Red Sea, there's a lot of variability from south to north, but the environmental conditions of the deep water of the Red Sea are very uniform and stable across the Red Sea, and they're characterized by very, very low oxygen levels. And as I mentioned earlier, then temperatures that never go below 21 degrees. So very warm waters, very low oxygen levels, and also very good conditions for calcifying organisms that elsewhere in the ocean cannot really grow below 600 meters depth. And we conducted the first assessments ever, I believe, in any ocean of the metabolic rates of deep sea corals to understand their functions, and be able to understand also how they are able to cope with the very low oxygen conditions of the deep water of the Red Sea. And very remarkably, we found that these deep sea corals are able to continue to survive even under complete absence of oxygen for at least 1 week. We were totally surprised, because most corals require levels of oxygen of about 2 to 3 milligrams of oxygen per liter to be able to survive. And these deep water corals are able to go without any oxygen at all for at least 1 week. So they're extremely tough and resistant, and there's no case like this reported anywhere else in the world. So your deep water coral reefs are truly unique and resistant, not only in their resistance to low oxygen, but we also tested their capacity to be able to grow in warmer waters. As I mentioned earlier, most deep water coral reefs elsewhere, corals can only grow to temperatures up to maybe 14, 16 degrees, but the corals in the deep water corals in the Red Sea are already growing at 20, 22 degrees. So we tested how much we can push temperature for these corals to be able to continue to survive, and we found very remarkably that they're able to survive to temperatures up to 33 degrees. So that is extremely surprising. It surprised us greatly, and it is by far the most thermal resilient deep water corals anywhere in the ocean. So very, very remarkable results from a biology perspective. We also studied the plankton communities that support the food web of the Red Sea, and we use that throughout the entire water column, from the surface down to 3,000 meters, using a device that was designed by Professor Susana Agusti, that is called Bottlenet. So it's a device that allows to collect samples along different depths while still collecting water samples with the Rosette sampling system. And with these surveys, then we found that there is a systematic change in plankton communities from the south to the north of the Red Sea, shifting from diatoms in the south, which benefit from the nutrient-rich waters that enter from the Indian Ocean through the Bab el-Mandeb, to dinoflagellates, which are more resistant to low nutrient conditions in the northern part of the Red Sea. We also found that about 0.6% of the carbon that is produced in the upper water sinks to the deep sea and represents an important sink for carbon contributed by plankton in the Red Sea. And also we determined that plankton in the Red Sea is extremely efficient in moving carbon up the food web, so that supporting the very remarkable densities of mesopelagic fish, which are small fish of about 2 to 4 centimeters that occur in the Red Sea in remarkable abundances, which is very surprising because we could not, before the expedition, understand how this nutrient-poor ecosystem could support those very large populations of fish. But now the work done on plankton then helps understand that the food web in the Red Sea is extremely efficient in being able to support fish biomass. So in fact, these mesopelagic fish, they migrate from the surface to the deep waters during the sunrise. So they come to the surface to feed at night, and they migrate down to about 600 meters during the daytime to hide themselves from predators. And you can see that in that image where the green-blue color is the density, the acoustic signals of the fish, and they move down, so they go down, and at some point they encounter the seafloor, and where these fish communities encounter the seafloor during the daily migration, then they are predated by a number of organisms. These have never been reported, so we have the first evidence that these mesopelagic fish, that are the largest stock of fish in the ocean with 30 times more biomass than all of other fish combined, are actually an important food source to the biological communities in the seafloor. And we are using now DNA analysis to understand what is the prey that these mesopelagic fish are living on. So it's mostly copepods and also other small plankton organisms. And we not only observe for the first time these mesopelagic fish alive, which had never been observed before, but we also have the first data on what are the food sources for these fish and the entire food web that keeps the Red Sea thriving and healthy. And as mentioned earlier, we also discovered that sharks in the Red Sea grow at greater depths than anywhere else in the ocean, and that is a consequence of the warm waters of the Red Sea, because it was recently published that sharks elsewhere, when they dive in deep waters, they cannot feed because they lose a lot of heat through their mouths and gills, so they cannot really penetrate deep. But in the Red Sea, because of the warm nature of the waters, then these shark species can penetrate much deeper than elsewhere, and also actively feeding. So this is one of the deep sea sharks that are feeding in bait that we place on the seafloor. So we have an abundance of observation of sharks feeding in the seafloor, and also predators, other sharks predating on smaller sharks in the deep sea, which is remarkable. And we assembled the largest data set of sharks in the Red Sea, understanding their distribution from south to north. And we have assessed also that the abundance of shark in the Red Sea is below what they should be because of hunting and fishing of sharks, which is also an issue that will need to be corrected to be able to lift up the populations of these predators that keep the food web healthy. We have observed for the first time the occurrence of very rare animals called sea elephants that live in the sea. They are only known dead. Nobody had seen them alive before. So these are the first images of these remarkable animals that have been very rarely reported from the sea. And our results not only confirm the existence of one new species, but also provide the first records of these animals living anywhere in the sea, and provide a revision to what we know about the distribution in the ocean. And we also observed for the first time this very remarkable animal that crawls on the deep sea floor that had never been also reported elsewhere in the Red Sea, but not seen alive. So these are very unique observations that we have been published already that improve our understanding not only of the Red Sea, but the global abundance of very rare animals, because if they are caught by nets, they are destroyed. So they're very difficult to be studied, and they have never been seen alive. As mentioned earlier, we have discovered two chimneys fields in the Red Sea, one just north of the Farasan Islands, which are very remarkable microbial towers that are skyscrapers that are formed over thousands of years of time by the activity of microbes feeding on the fluids that are released by the crust of the Red Sea, and that also harbors remarkable biodiversity like a crab that grows inside these chimneys that are devoid of oxygen and full of sulfide. And these crabs are able to survive there because they have bacteria on the gills that detoxify the fluids that come through the gills, and it's very remarkable that these animals can survive in that very harsh environment. We also found another field of chimneys, this time not inactive chimneys, in the northern Red Sea. So those are two new discoveries, and again, they not only are important in terms of mapping unique biodiversity with remarkable capabilities, but also inform of the activity of the seafloor in the Red Sea, as also the collection of volcanic rocks from the depths of the Red Sea also provide. So we collected more than 100 volcanic rock samples from the deepest waters of the Red Sea that already inform about the origin of the Red Sea, volcanic activity in the Red Sea, but also contain remarkable biodiversity, including a new family of corals that live in these rocks in the deep sea. We are also finding, using the combination of standard taxonomy and molecular methods, remarkable abundance of new species in the Red Sea across all taxa. So we're contributing to the inventory of species in the Red Sea, even with polychaetes. And we're also looking at the impact of dust deposition in the Red Sea. So there's about 5 major dust storms every year affecting the Red Sea that deliver about 6 million tons of dust into the Red Sea. And we did not really understand well what is the impact of these dust storms on the functioning of the ecosystem, but we have been documenting the changes in dust delivery to the seafloor of the Red Sea over time, and we found an increase in the amount of dust that is deposited that also is a consequence likely of the expansion of deserts around the Red Sea, and also calls for improvements in regenerating land and combating land degradation. There's going to be a major focus of the Kingdom of Saudi Arabia this year, hosting COP16 in Riyadh in December. And we also explore marine litter in the seafloor. So we sample a lot of areas in the seafloor of the Red Sea down to 3,000 meters, and observe that the litter in the seafloor of the Red Sea changes greatly in abundance, and is actually concentrated along the main path of shipping along the Red Sea. So the vessels that cross the Red Sea from the Bab el-Mandeb to the Suez Canal, unfortunately, despite the MARPOL Convention, it seems like the mariners on those vessels keep disposing of cans and bottles and other litter into the Red Sea. So it's really littered in the center of the Red Sea, and we have recovered mostly plastic litter and also metal like cans and glass, ceramics, and other litter from the center of the Red Sea. And usually in most oceans, most of the litter is near the coast, and then the density of litter decreases as you go into the offshore. Here's the other way around, because there's very limited inputs of litter in the shoreline of the Red Sea, and most of the litter comes from the vessels that are crossing the Red Sea. So I believe there needs to be work done with the IMO to be able to improve the compliance of the vessels that cross the Red Sea with the MARPOL directive that prevents littering and tossing cans and bottles and so on from the vessels into the water. So then, as I mentioned, one of the core pillars of the expedition was to conduct biodiversity end-to-end, and in the 21st century, we will not assess biodiversity in the way we did in the past. So now we will use high-throughput sequencing approaches to do that, and we have sequenced, I think, about 4,000 samples from the Red Sea to generate inventories of biodiversity using what we call environmental DNA, which is just a sample of particles in the water that has remnants of cells and materials of every species that was around. So by massively sequencing that material both in the water column and sediments, then we can get a full list of species that are present. So now we are retrieving those lists of species. For many of them, we don't even have a name because every species is detected, and we know that there are a lot of species that are present in the Red Sea that have not been formally described. So we can guess what their taxonomy is, but there is no name for these species. And as we move forward and we develop, together with the National Center of Wildlife, a barcoding program to be able to attach sequences to species in the Red Sea, we will be able to unveil a lot of biodiversity for which we have the DNA but not a name yet. We also collected and sequenced about 300 samples for metagenomes, so that means that we have sequenced all of the genes that are present in the organisms. And as I mentioned earlier, the catalog of genes in the ocean now has a strong commercial interest and industrial interest and potential. And of the many millions of genes that we are reporting, probably a very large fraction will have industrial applications eventually. So to be able to process this very large amount of information, we have challenged the National Center of Wildlife with this project because we are generating, the data that is being generated is in the order of petabytes. So you need supercomputers to be able to analyze this data. We're using Shaheen 2 and in the future Shaheen 3, which is Cow supercomputer, now ranked 20th most powerful in the world, to be able to analyze all this data and generate genomes for species, generate a catalog of genes, and also assign taxonomy, so we have a full inventory of biodiversity from bacteria to whales. And we have found so far about 6,300 species of higher organisms through our sequencing efforts. But the most important component of the sequencing effort is that we do not generate only a baseline of the current biodiversity of the Red Sea, but when we sample the seafloor and the sediments in the seafloor, and we analyze DNA at different layers, we're actually going back in time. So we are assigning the date, the age of the sediment, and then sequencing different layers of sediment. So we're going back to the biodiversity of the Red Sea in the year 1800. So this shows that over time, from 1800 to date, there's been an increase in biodiversity in the Red Sea, in diversity, but we need to unpack this information by looking at targeted species. So my colleagues and I are looking also at the species of interest, not just the total biodiversity pool, but looking at the trajectories of sharks, of sea turtles, dugongs, and other organisms of conservation interest, so we can have baselines of what was the historical abundance of these species in the Red Sea. So we are now busy also reporting our results so that we don't keep them just for ourselves, but the National Center of Wildlife from the beginning wanted for all of the research done in the expeditions to be publicly available to scientific publications. So this is our timeline of publications, and we are now just scratching the tip of the iceberg of the results a year and a half after the expedition, because we are generating, as I mentioned, petabytes of data, so it takes a lot of time to process those, but we hope that by the end of this year, inshallah, we'll reach to about 70 papers published in the international media, international scientific journals, so that our target is by the time we get done with all of the samples from the Red Sea Decade expedition will probably be around 200 scientific papers, and that will be a fundamental block of knowledge on the ecology and biodiversity and potential to support the blue economy of the Red Sea. So thank you for your attention, and I again congratulate the National Center of Wildlife for leading the expedition and also organizing this amazing symposium, and we hope to be again with you maybe in a year again to report even more discoveries. Thank you.
Thank you very much, Professor Carlos, for the very interesting and important discoveries that have been presented. Our esteemed guests, we would like to now invite you for a quick lunch before we commence with the rest of the speeches and talks. Thank you very much.
We call the Red Sea the baby ocean. It contains all the features of any ocean. Red Sea is very unique that you need to understand it well. And this is what we did in this cruise. And this is what we call the Decade Expedition for the Red Sea.
What we're doing in the Red Sea Decade Expedition is bold, and it has not been done before. I'm not aware that any nation has tried to produce an inventory of their marine biodiversity through an approach based on environmental DNA. To be able to do that in a research vessel is even more unprecedented.
One of the many advantages of eDNA is that we do not rely on the organisms actually being present at that very moment. Since we're observing wildlife, we cannot predict where they are going to be and how many are there going to be.
After 8 months of planning for the Red Sea Decade Expedition, lanternfish were not in my mind, but when I boarded, I looked at the echo sounder and I saw that distinct signature of the animals moving up and down. Just by placing the ROV at the right depth at the right time of the day, I realized that we had a major discovery. We could, for the first time, calculate their density and even observe their behaviors. So those little fish, they're important because they bring me hope that we haven't yet broken the ocean.
We cannot comprehend life without plankton. They are so simple in their form, but they are so complex in what they are doing. We're trying to have a good understanding of how the plankton community in the Red Sea, what is the abundance, the distribution, and the biomass. The Red Sea is very unique in terms of temperature and salinity and low nutrient. What makes the plankton in the Red Sea very unique is that they are very efficient in their utilization of the nutrient in the area. Shark.
Yeah, what?
Here.
Oh, great! The great shark.
Shark.
Great shark. It was amazing because we caught a lot of sharks, at least 4 different species, and one of them is the scalloped hammerhead, and it's a critically endangered shark.
Our research that we are doing now can be in the biology classes tomorrow in our school.
The coral reef is well developed all along the Red Sea, which is a very, how can I say, it's a very weird basin because it's an enclosed basin and it has the highest salinity, and also the temperature is very, very high compared to other ocean. We have species that can tolerate thermal stress, and we are trying to understand what is different compared to other corals from other regions.
We have the best 2% coral reef in the world. The genome in the coral reef is resistant to high temperature.
So understanding how ecosystems within the Red Sea are so good at dealing with these conditions and thriving is incredibly interesting from a research perspective, and understanding how the ecosystems flourish in this sea can provide us with valuable information about ecosystems around the world.
When we discovered that brine pool, we found that in addition to that body of highly saline water, we found a series of chimneys that are emerging from the sea floor along with microbial growth that are releasing methane and oil and form a landscape which I believe is quite unique in the world's ocean.
The Red Sea is a unique ecosystem. It's mostly an enclosed body of water, and what's happened over thousands and thousands of years is that the species that are resident in the Red Sea have become unique in their own way, and there are a number of species that aren't found anywhere else. The plan during the expedition is for us to use the helicopter to conduct standardized surveys along the entire length of the Red Sea.
We had some amazing discoveries today. A couple of mother and calf whale pairs. It's probably one of the greatest discoveries for me so far on the entire journey.
8 o'clock, 300 meters back. A patch is splashing just down here. Okay, somebody find me a whale.
What's that?
That is
A whale! Oh, thank God.
Just amazing. Lucky.
Studies will never stop as long as we are, as a National Center for Wildlife, working for the conservation. Studies will continue and monitoring and observing. Our main goal is the conservation and keep the biodiversity as the biodiversity is very important to our life.
والآن حضورنا الكريم يمكنكم الانتقال لتناول وجبة الغداء. Thank you, esteemed guests, and now we would like to have our lunch. Dear guests and participants, we would like you to have your seats. We will commence with the rest of the talks shortly. Welcome back, dear guests. I hope everyone enjoyed their lunch break. And to continue with the Red Sea Decade Expedition Symposium, I would like to ask Dr. Nicholas Pilcher, marine scientist, expert, and megafauna researcher, to present a paper on Red Sea megafauna.
Good afternoon. Buenas tardes. It's a great pleasure to be with you this afternoon to present on some of the findings from the Red Sea Decade Expedition. As many of you know, I had the pleasure to lead the megafauna surveys on Ocean X, and much to most people's jealousy, I got to spend immense amounts of time up in a helicopter counting wildlife. All of these talks require the obligatory turtle photograph, being as today I'm going to focus on sea turtles. So now consider yourself turtle introduced, and they also require the obligatory pictures of boats and helicopters and things like that. So that's the end of the photos. From now on, we'll discuss a little bit more in detail on some of the findings. What I'd like to do this afternoon is focus entirely on the green sea turtles and the findings that we have from the data from the Ocean X surveys on that. As a little bit of background, this was the planned survey tracks. All of these tracks occur in relatively shallow waters, and they cover most of the reef ecosystems of the Red Sea, the Saudi Arabian Red Sea. As you can see, we plan to do somewhere in the region of 14,000 kilometers worth of flights, but due to a number of logistics challenges and things like that, we ended up cutting back some of the surveys to about 9,000 some odd kilometers. Part of that was because I was taken off the surveys after phase 3 due to some other projects that the center had going on at the time. And then in terms of actual flights with the helicopter itself, as you can see, we did something like 9,400 kilometers worth of flying over the 3 survey periods. So it's quite a lot of detail. Just to show you in a little bit more detail, this is the Al-Wedge Lagoon, and these were the planned tracks for the Al-Wedge Lagoon. And then you can see very clearly that the helicopter tracks followed those very, very precisely with a lot of accuracy. And in the places where you see the little loop-de-loops, places like these are where we had sightings, and we asked the helicopter pilot to backtrack so that we could record those sightings and make sure that we had the species and the counts correctly. So moving on from Google Earth and trying to get into a little bit more detail with the statistics, the survey design with the helicopter was, the survey path was designed to cover a width of 400 meters. And so eventually we managed to survey roughly 3,500 square kilometers of the Red Sea out of a planned shallow water total survey area of about 47,000 square kilometers, which works out at an average, also just zooming in again back into the Al-Wedge Lagoon to give you an idea of how that survey area was designed. You have the tracks that go over the shallow water, and then overlaid on top of that is a broader shallow water habitat. And so the transects with the aircraft are a strip transect from which we can extrapolate data across to the larger shallow water habitat. And as you can see from here, the overall average survey strip transect coverage represented about 7.5% of the shallow water habitat that we aim to survey. During that time, we counted a total of 380 sea turtles, so 380 individual turtles during these sightings. I also want to draw your attention to two specific examples that I think are worth just noting from the records. One, if you can see in this slide, you can see the little squares, and these squares were undertaken during phase one, or leg one of phase one. Dr. Carlos was on board at the time and I was not, and we needed to design a survey that would cover deep water habitats in a statistically valid manner. And so these two squares are just to give you an idea that instead of parallel transects, there are other ways to do this work. And the second example I want to draw your attention to on the right of your screens, notice all of the zigzags in and out, back, across, here there's a pointer here. So these points over here, these are the locations of the Ocean X vessel where we had to return every time to refuel and go back out again. And the reason I point this out is because this is an incredibly inefficient way to do surveys. And I say that because the distance traveled to get from the vessel to the beginning of the survey line and back again was nearly 2/3 sometimes of our fuel availability. So in many ways it's very inefficient, and it would have been a lot more helpful, obviously, for the vessel to be closer. Of course, the downside is the vessel was doing 1,000 other things at once, and so it's not always possible to get the best of all worlds. But moving on to the actual data, this graph shows a relationship between the depth at which each individual turtle sighting occurred and the distance from the reefs. And all you really need to take away from it is nearly all of the turtles were seen up close to a reef in shallow water, clustered up in the top left side of your graph. So there was a significant correlation between the depth and the distance from reef habitats, and these were calculated using publicly available bathymetry charts and GIS distances from the nearest reef. The next thing that we found is that there was a significant difference in the distribution of sea turtles by age class. This is the distribution of turtles across all of the sightings that we had in the Red Sea, but then this one breaks them down by juveniles and adults. And you'll notice that all of the juveniles are seen in extremely shallow waters, with very, very few cases of juveniles being seen out in deeper waters. And in the cases where juveniles were seen in deeper waters, they're very possibly crossing from one area to another. So putting these numbers into perspective, total survey area, roughly 47,000 square kilometers, flight coverage about 3,500, total turtles 380, and if you extrapolate that 380 turtles across the entire zone, the orange zone that I showed you a little bit earlier, we arrive at an estimate about 5,000 sea turtles that could have been in that area. However, the challenge in interpreting that data point is that not all turtles are available to be seen at the same time. Right? Turtles spend a lot of their time underwater, and if they're underwater, we're not going to see them. So based on the proportion of time that turtles spend underwater, and there's a slight variation between juveniles and adults, you'll notice that about 222 of the sightings were in shallow water, where there's a higher probability of being seen, and 158 of those sightings are in deeper water, where there's a lower probability of being seen. Then the next thing that needs to be corrected for is the perception bias of the observer. And what that means is that as the helicopter is flying along, each individual observer might not be able to see every single turtle that goes by. So if we take the estimated 5,000 sea turtles and we correct that for availability error by depth, and we also correct that by perception bias from the observers, we arrive at an estimate of roughly 65,000 sea turtles for the orange zone that I showed you in the earlier graph. 65,000 sea turtles. It's pretty impressive, I think, right? And that with confidence intervals somewhere between 50 and 100,000. So those are numbers of turtles. Now, in the first ever survey of the Red Sea conducted by Miller in 1987, he never actually produced an estimate of this magnitude, but he did have a figure of roughly 1.54 sea turtles per square kilometer, and ours was nearly 10 times higher than that. Right? This is good news for the Red Sea. This is incredible that the earlier estimates of abundance are a tenth of the estimates of abundance today. Now, this is actually not surprising to me. Two reasons. One, Jeff's work in the 80s was a lot more limited. And two, we know from work of people like Takashimada and colleagues that the numbers of nesting turtles have grown by 3 times as much. So it's not surprising to see increases in the numbers of sea turtles, not to mention the fact that there was extremely robust methods and extremely detailed effort during the Ocean X expedition that Jeff unfortunately wasn't able to do back in the 80s. So I think this is an amazing number, and it's something that we'll come back to shortly. In terms of distribution, this is the overall distribution, just in a very generic heat map, an overall distribution of green sea turtles along the Red Sea. But I also want to point out something else, that this graph here, this is the distribution of just the adults. Right, so this does not count any of the juveniles, and this is the distribution of juveniles. How amazing is that, right? That all of them are up there. And if we zoom in a little bit closer, this is the headland by Rasbaridi, with the cement factory just off the second of those bays. And that is the heat spot for nearly all of the juveniles that were found in the Red Sea. So this survey has managed to conclusively identify the largest and most important juvenile foraging ground for green sea turtles in the Red Sea. It's probably not going to come to a surprise to some of you who work on seagrasses in the area, but it's quite impressive to think that this is where all our juvenile turtles come from. And if you're going to protect anywhere in the Red Sea for sea turtles, this should be one of those places. So those are the numbers, and I'm trying to keep it brief. Those are the numbers, and I want to try and give you some thoughts in terms of what are the management implications of knowing those things today. So first of all, we don't need to undertake surveys out into deep water if we're counting green sea turtles. We know that they all occur in shallow waters. We probably could limit the extent of our survey area and do less work and collect similar, if not better, amounts of information. Critical habitat for juvenile turtles lies in waters less than 10 meters, and particularly in that area south of Rasbaridi, southeast of Rasbaridi, is a critical area for green turtles in the Red Sea. This survey estimated approximately 65,000 green sea turtles up and down the shallow waters of the Red Sea. And one thing I want to caution you is that this is a point in time. And the reason why this is important, if we go to these graphs, on the left you have nesting data for Hawaii, and on the right you have nesting data for a place in Malaysia called Sarawak. The important point to take away from this is notice how the population on the left goes up and down through time. And every single one of those points is a valid point in time. But if you would agree, the population seems to be on a generic upward trend. If you look at the trend on the left, the trend for Sarawak dropped dramatically in the 30s down to the 60s, but has remained stable ever since. The two key points to take away from this are that for sea turtles, a single data point does not tell you very much. You need multiple data points over a long period of time to be able to identify trends. And so our 65,000 sea turtles for the Saudi Arabian Red Sea shallow water habitat is but a point in time. And it's the repetitive conduct of surveys like the ones that took place during Ocean X that would give us that sort of data. The increases that we see in numbers of turtles or density of turtles that Jeff Miller recorded in the 80s and we've recorded 30 years later, 40 years later, mirror in many ways the findings of the nesting increases over time that have been recorded in the Red Sea. These surveys, because of the nature of the surveys and the way the data was collected, the perception bias and the availability bias that we have for this particular work comes from similar work elsewhere. It doesn't come from our own work. And so future surveys are required, particularly to try and get an understanding of availability bias. And one of the best ways to do that would be to conduct some standardized surveys where you had turtle silhouettes at different depths. And that would give you an availability bias based on depth, and it would give us a much better estimate of abundance in subsequent surveys. The Khor south of Ras Baridi is a critical development bottleneck for green turtles in the Red Sea. And if we lose this habitat to development or to any sort of damage, we risk losing a very, very valuable resource in the Red Sea. And looking into the future, and particularly thinking about issues like climate change, in this area in Ras Baridi where there's all of the juveniles, we could implement studies of in-water sea turtle populations that would give us real-time data on the impacts of climate change. This is work that I've managed to undertake in Qatar and in the UAE and in Iran over the last decade, in other parts of the world as well. But a lot of the times when we're looking at what is the impact of sea turtles, there are people that use models to try and predict what might happen. But if you could go into a juvenile habitat where you could sample animals and actually get an exact record of what was the proportion of males and females, that would tell you what's happening in the ocean. And if you could conduct that over time, you could get a very, very clear understanding of what the impacts of climate change might be. The reason why it's more important to do that than to sample eggs or incubation temperatures on beaches is because in the Arabian region, temperatures have been warm for a very long time. And so just knowing that sea turtle eggs are incubating in warm temperatures doesn't necessarily mean that it's going to lead to a feminization of the population. In order to do that, one requires getting in the water and actually sampling in-water turtles. And for the very first time, we now know where that site is. And with that, thank you very much.
Thank you very much, Dr. Nick. Next, to be presenting on biology of deep sea corals in the Red Sea, I would like to ask Dr. Alexandra Stuckbauer, a research scientist and marine scientist from KAUST.
Thank you, Omar, for the kind introduction, and my thanks go also to the National Center for Wildlife for the invitation to speak to you today. After a quick introduction on deep sea corals, I will present 4 different projects we're currently working on the deep sea corals. This includes environmental conditions, onboard incubations we conducted on Ocean X, oxygen drift experiments in the lab back in KAUST, and some observations we observed. Coral reefs are also called the rainforest of the sea and are a very extraordinary ecosystem that harbors remarkable biodiversity of marine life and is also called a biodiversity hotspot. It's a very important ecosystem for feeding and breeding of different invertebrates and fish species, and is also important for fishing and livelihood of nearly 1 billion people worldwide. And contrary to the shallow reefs that are down to 30 meters and are found in the tropical and subtropical regions, deep sea corals start around 150 to 200 meters. Usually the deep sea corals, compared to shallow reef corals, don't have the symbionts, so they depend on other forms to feed. As you can see here in the picture, they usually grow in the direction of the current, as they are filter feeders. They're also the synonyms to cold water corals. As we already heard today, usually these deep water corals are between 4 to 14 degrees, but in the Red Sea they are found at around 21, 22 degrees. They're also pressured by different threats, ocean acidification, deoxygenation, among others, and also bottom trawling and fisheries. About the conditions where they live, previously publications showed that they live around 2 milligrams per liter oxygen and 21 to 22 degrees temperature, and also in high salinity around 40.5 PSU. To adapt to these conditions, they have a very low metabolic demand. We have gorgonians, so soft corals, and sclerotinian corals. Gorgonians we found, or we observed on the vessel, whip corals and fan corals. As I mentioned before, they are filter feeders growing with the currents, and they capture plankton and other microorganismic organisms that they feed on with their polyps. And they also provide habitat and refuge for fish, crustaceans, and echinoderms. One of the remarkable sclerotinian coral is Rhizotrachus typus, which was found in also a very high oxygen range from 5.5 to down to 0.5 milligrams per liters. For the environmental conditions for this project, we used the ROV data of the CTD, where we collected data of oxygen, temperature, salinity, among others, and we measured on 114 ROV dives along the Red Sea coast, from both the deep sea till the shallow reefs on the different phases of the expedition. And we found and can confirm the data we found previously, that there's this ocean minimum zone between 150 to 500 meters, and then oxygen is pretty stable after around 750 meters down to the deep of nearly over 2,000 meters. Similar for temperature, it reaches at around 200 meters, the 22 degrees, and then pretty stable down to the deep ocean. And similar for salinity, it's going up 40.0 and also stable. These data are, for example, used for a niche modeling approach we are using to determine which conditions corals prefer for the absence and presence. And for that, we will hear later more from my colleague Megan Nolan. To the onboard measurements, we collected different corals with the ROV and the submarine. Here, for example, you can see the slurp where we collected them. We put them in individual containers and kept them in the dark, and also labeled and took pictures of the specimens. For these components, we fragmented the corals, so we cut them into tiny pieces, and then we put them in small chambers. Here you can see our system to measure the oxygen consumption during a more or less 1 hour incubation. And here I show a quick video how we push them to this. We're working with red headlights as we stayed in the dark, as this is the natural conditions of the corals. In total, we measured 67 incubations of 82 samples. We also measured dry weight and ash-free dry weight to standardize the data, and we did identification to see which species they are. What we can see is that we had higher respiration in the north of the Red Sea, except these 2 outliers. And also with the depth profile, the results are mimicking these oxygen trends in the deep. If we look on the taxonomic groups, we can see that these highest variability and highest respiration rates were with the genus Acanthochargia, and we had really low ones with Nicella. But here we are still working on the final results and also run the statistic tests. In the next part, I'm talking about a lab experiment we did back in KAUST. Lead scientist is Ms. Jacqueline Alba Garcia, who is a PhD student. For this, we wanted to see how the oxygen concentration is affecting the organisms. And this is very important to understand the deep sea corals' physiological responses and also to their resilience to climate change. For this, we used, again, the Rhizotrochus typus. We used 50 specimens that were collected from 146 meters to over 730 meters depth. On board and back in house, the first were kept at 8 milligrams per liter oxygen, 22 degrees, and a pH of 8.2. And here on the map, you can see that we covered Gulf of Aqaba down to the south, the specimen. We used our gas system in KAUST, where we combined gases, nitrogen, oxygen, and CO2 to reach 22 degrees, and from the pH 8. And we transitioned the oxygen down to 0. Originally, the plan was to stop the experiment after 14 days. However, they were really resilient and survived. So we extended the experiment. Besides oxygen flux measurements, we also did samples for transcriptomics and metabolomics. Here again, you can see a close-up of the corals in the chambers. The respiration we measured at the beginning and after 7 days of them being in anoxic conditions and at the end. And you can see that the respiration was going up as they were more stressed. Remarkable was that they survived 7 days in anoxic conditions. And the first dead specimen we had on day 23 of our experiment, then 2 more dead on day 24. And the last dead was on the day 27, so we were able to run another respiration at the end. Some observations we did on the boat is, as I mentioned before, the deep sea corals don't have the symbionts, so what are they actually eating? As we saw before, lanternfish are migrating from the surface waters down to the bottom, down to around 600 meters. So here we have sunrise where they start migrating, and sunset they migrate back up. This is very important for the benthic pelagic coupling and also the transportation of the nutrients. Here I show a few videos. You will see here the fish coming and getting trapped in the tentacles of the coral. This coral had 4 to 5 specimens already trapped in its tentacles. And in the last video, you can see how the tentacles are already retracting towards the mouth of the polyp as it's digesting the lanternfish. In total, we noticed so far 13 observations. One of them was also done by Professor Benzoni and her lab on a specimen that was collected. And here you can see the eyes, and the coral has like 3 to 4 lanternfish inside. Other observations include anemones. Again, you can see the trapping of the lanternfish and the devouring of the fish, but also lobsters and crabs and fish. To conclude my presentations, the Dead Sea corals are very well adapted to their environments and have a very low oxygen demand. And these videos shed first light on the behaviors of these deep sea corals. And with that, I want to thank again NCW, Professor Carlos Duarte, Dr. Corban. It was an honor to be part of the expedition, and thank you very much.
Thank you very much, Dr. Alexandra. And next, we would like to ask Professor Susanna Agosti, Professor of Marine Research Scientist, Professor of Marine Science, KAUST, to talk to us today and present about plankton communities in the Red Sea.
Okay, thank you, Mark. It's my pleasure now to introduce the work we run about the studies on plankton communities during the expedition. So as Professor Carlos Duarte introduced this morning, well, during the expedition, we focus in the microplankton. We have to consider that these tiny microscopic organisms, they are very important because they are responsible for very important functions in the ecosystem. So we have the opportunity with the Recidica expedition to sampling the deep waters and also the deep waters, the water column, the total water column. And this is what we did. And well, the Red Sea is not a very deep sea, but maximum depth is 3,040 meters. So we have 3 of the more important layers in the oceans, which is the epipelagic, which is the illuminated layer, the mesopelagic zone, but also we have some areas with the bathypelagic. So we were able to sample the all 3 layers in the expedition. To capture the plankton, we used this bottle net, which is a special net that you can choose the depth when you open and close, so we can precisely select which depth we sample. So what we got, well, we did this work in the Ocean Explorer, and what we got is an integrated sample from the different layers. No? So from the surface to 180 meters, from this depth to 1,000, which is the mesopelagic, and below to the bottom, which is the bathypelagic. And what we catch with this net is this kind of plankton. So we catch the large photosynthetic organisms, the large phytoplankton, and this is mostly composed by these large diatoms and dinoflagellates. We were also able to catch the zooplankton, and in this case, the small fraction of the zooplankton. We catch also the large colonies of the cyanobacteria trichodesmion, which is a key species. Also, we catch the aggregates of plankton and detritus and the fecal pellets, components that are important for the export to the deep ocean. So we have 3 major goals. The first is to explore the biodiversity changes geographically, so across the Red Sea, and also in the water column, so from the surface to the bottom, and then evaluate what is the phytoplankton export, the primary producers export to the deep sea. And by this, then we maybe have the opportunity to also explore what is the relevance or quantify the biological carbon pump in the Red Sea. So the biological carbon pump is an important process. Is the biological driving capture of atmospheric CO2 that can be implied as sequestration to the deep ocean. So the phytoplankton captures the CO2 from the atmosphere, which is dissolved in the oceans. This produces different organic matter. This organic matter is spray and is transported to the food web. But some part of this primary production don't go to the food web, but by different processes is sinking and is exported to the oceans. So is this sinking particles, this sinking production, the one that will be entrapped? And when going very deep, below 1,000 meters, we consider that is sequestered. It's no capacity to return to the atmosphere, so it's a net sequestration of CO2. Okay, so well, we examined the phytoplankton communities in the expedition, and for that, I got the help of Masen Safia, which is a laboratory, my technician in my lab. And well, this is a pattern that we know. In the south of the Red Sea, we have much more abundance than in the north, but this is quite relevant that we found a very large variability in that. So in the south, we can catch millions of cells per cubic meter, but in the north, this decreased to even maybe 7,000 cells per cubic meter. And what we observe is that all these changes in the abundance of phytoplankton cells is due to the presence of diatoms. So as diatoms increase, the total abundance increases. Okay, we also can describe well this pattern in the proportion of the community, the changes in the community with latitude. This pattern has been already suggested, but never shown so clearly, as we have the opportunity to show here that diatoms dominate this community by 90% in the south. Above 22 degrees north, these start to change, crossing that with the presence of dinoflagellates that can become the dominant in the very north of the Red Sea. So this is an relevant change in the community across the Red Sea. During the expedition, we were also able to identify very large blooms of phytoplankton. And we observed that in the north of the Red Sea, especially around 26, 27 degrees north. And well, those blooms were also visible from the air. So those images were captured by the helicopter during the expedition. So one of these blooms was dominated by Coclodinium, which is a dinoflagellate, which is considered a harmful algae. So it is difficult to identify the species, but some species of this Coclodinium are harmful algae. They are producing chemicals that can kill other organisms. So it's a concern that we have these blooms in the Red Sea. The other bloom was dominated by the cyanobacteria Trichodesmium, which is considered a keystone species. Is a species growing in tropical waters, and it's important because it's fixing nitrogen from the atmosphere. So this is a source of nitrogen for the ecosystem, because it can leak between 30 to 50% of this species nitrogen to the environment. So it's a source of nitrogen. We also observe other key species. And in some of the colonies of the diatoms, especially from the south, we identify this another cyanobacteria, Richelia, which is an endosymbiont. And this is also fixing nitrogen. So these diatoms are receiving nitrogen for the presence of this symbiont. So this is also explaining why they can also grow to high abundance in the South Red Sea. We also observed what happened in the deep waters, in the plankton, and this is exported. So we observed that in the mesopelagic layer, we have a composition that is mimic to the surface. Of course, these cells are coming from the surface, so we see diatoms again to dominate and in the south and decreasing to the north, but we still have in the north a higher proportion of diatoms in the mesopelagic than dinoflagellates. This pattern was more drastic when we observed the few stations, because we have shown only where we had bathypelagic waters. So clearly, as we go down, the community is getting dominated by the diatoms. And this is consistent because the diatoms, they have this siliceous wall with this heavy, and this will increase their potential sinking. Also, it is described that they have a highest tolerance to the hardness. So we found that the diatoms are key players in the Red Sea in carbon sequestration, and this is described for other ecosystems. So the biological carbon pump, well, phytoplankton, of course, is taking this CO2 from the atmosphere, compared to biomass, but only few of this production will be exported. And this is what we found, because we found that in the mesopelagic, the abundance, the biomass that is exported from the phytoplankton is only 4% of the biomass in the surface. And if we go to the bathypelagic, what is the real layer with this occurring carbon sequestration, this is very small. This is only the 0.6% of the epipelagic biomass. However, this is not enough to really evaluate what is the efficiency and the capacity of the biological pump. So my PhD student, Wan Liu, was participating in the expedition, and she was measuring the primary production by this microplankton in the surface. And also the goal is to calculate the efficiency of the biological pump will be related to how much carbon is transferred to the bottom relative to the primary production in the surface. So, well, the Red Sea is in general an oligotrophic sea, so we expect that this carbon will be not very high because the production is not very high. But this is one of the warmest sea waters, so we have high temperature in the meso and bathypelagic layers, so we don't know really what is the capacity of the bacteria growing in this layer to decompose this phytoplankton and remineralize and affect the efficiency of this carbon pump. We confirmed that we have diatoms, that these represent a high potential for the export. So we want to test what is the quality of this exported phytoplankton to the deep. So, well, even that these meso and bathypelagic layers, this plankton is not receiving light, we expose to light. So we collected from the deep dark ocean and it exposed to the light, and then we quantified if they were able to make photosynthesis. And what we found is that, yes, they were able still to make photosynthesis. And of course, the quantity of photosynthesis we can get from that was very small, but we observed it in both the mesopelagic samples and in the bathypelagic plankton collected in the Red Sea. We tested with another method, with pump fluorometry, which is testing the efficiency of the photosynthesis, and this get how effective is the photosynthetic apparatus. And we observed that, yes, both mesopelagic and bathypelagic phytoplankton, they were efficient. Of course, efficiency was lower compared to the surface, but still they can photosynthesize. And that this means that these cells might be sinking fast to the deep, because if you think that this is a tiny organism with some micrometers, and they have to transport from the surface to 2 kilometers to really reach the bottom just for gravity, they will require more than a year. So this kind of cell for a year in the dark will be no survive. So this implies that we have a very fast sinking processes in the Red Sea that are moving faster distance to the bottom. And this implies that also the bottom of the Red Sea is receiving good food, still good products, not only the critical products with very few value for nutritious. Still, we have more data that we still are processing. So we are analyzing also in the samples the bacterial production, so the capacity to remineralize the carbon, the bacterial abundance in these particles that are sinking, what is the particulate carbon, okay, the carbon content, and also we also are testing in the phytoplankton, which we found some to be still alive. Well, what is the rate to death when they are exposed to dark, okay? Also, we will complement, and we are still working in this data. We have all the results, but we did not analyze yet what is then the particulate carbon that we can collect in the sediments. Especially relevant for this process is the deep sediments, the sediments below 1,000 meters depth. And also, what is the bacterial production and capacity to remineralize there. We take those samples from the cores that were sampled with the ROV in the Ocean Explorer. Another part of the team, led by my postdoctoral researcher, Zina, Chao Shu, which is a laboratory technician, they are quantifying the abundance and size of the depths. So the transparent isopolymer particles, well, this is the organic particles of special polysaccharides that are produced by both phytoplankton and bacteria, but those are sticky. So when these people are producing, they favor the agglomeration and increase the size of the particles, and they will favor the fast sinking of the particles. So while they are exploring with some image analysis of microscope samples after staining, because they are transparent, so you can stain and then observe. So they are doing in a series of samples from the expedition. Just finally, I want to add the work that is done by Afra Alotmat, which is a PhD in my lab. So Afra is studying the microbial loop in the Red Sea. So when I say that a part of the carbon is sinking, but the oil is going to the food webs, so the oligotrophic systems, the microbial organisms are very important because they are transferring most of the production to the rest of the food web, including the bacteria. So the bacteria is eaten by other organisms, and then they are using this as a food. So, well, this is the microbial loop. So we were exploring Afra at the study from the Red Sea Decade expedition to previous studies she was running. And well, what we found is, well, she was using a stable isotope of carbon, the 13 carbon, which we use as a tracer. So we use this in glucose, the bacteria will take, so we can quantify the heterotrophic production by bacteria, and then we will see how we can find this tracer in the upper organisms that are eating the bacteria and transferring to the food web then. So we found, yes, that the transfer of, we quantify how much heterotrophic production has been transferred. It is 70% higher than the bacterial production. And well, we confirmed that if you remove the grazers when exploring this, in fact, the bacterial production can increase more than 50 times. Also, what we observe is that this transfer of carbon is dependent of temperature in the Red Sea, so it is increasing with increasing temperature. So this is confirming the relevance of the microbial loop as in the food webs of the Red Sea. AFRA also is exploring other metabolisms. So we classify the organisms as autotrophic, those that make photosynthesis, and heterotrophic, those that don't, and use the products from the autotrophic, but this is not so clear. So there are some bacteria that is able also to uptake CO2 and use in a heterotrophic metabolism, and there is also bacteria that use light, not for photosynthesis, but for improve their energy. So bacteria was testing if this process was relevant in the Red Sea. AFRA was testing if this was relevant in the Red Sea, and we found that this is not. This photoheterotrophic production was not very important when we compare with the regular heterotrophic production. But probably this process is increasing in the areas where production is higher. Something to continue to explore. I just want to highlight the talk of my collaborator, PhD student, Said. He was participating in the LSEC, and he's studying another aspect of the microbial communities, which is when they are part of the microbiome. So he's exploring the microbiome in the macroalgae, and he ran this study in both the LSEC and the Ocean Explorer. And I invite you to attend tomorrow to his talk. And then I just would like to thank you for your attention. Thank you.
Thank you very much, Professor Susanna, for that interesting presentation. And next, we would like to ask Dr. Carlos Preckler, research scientist and marine scientist from KAUST, to give us a presentation about eukaryotic diversity of the Red Sea.
Hello. First of all, I would like to thank to National Center of Wildlife for organizing this event and to invite me to talk here. I'm going to talk about the eukaryotic diversity across the Red Sea. And I think it's super good that we had previously the nice presentation by Professor Agustí, because I think they match very well. And even a lot of things that she has been talking about, I think can help me understand a lot of my data. So the first thing, I would like just to explain what is an eukaryote, because maybe most of you already know, but I'm going to talk about all the time about eukaryotes, so I would like to be sure that all of you know what is an eukaryote. So the eukaryote constitute the domain of the eukarya, and they are organisms whose cells have a nucleus. This is one of the main important features of these organisms. And they range from the micro cells to the animals like the whales. So they accomplish a big range of biodiversity, as you can see in the right panel. So they were formed like merging one prokaryotic cell, so one archaea, and another cell that is aerobic bacteria. So after that, there were a second merging with photosynthetic that they were adding the chloroplast, and they form the green plants. So at the end, the eukaryotes range all animals, plants, fungi, and a lot of unicellular organisms. Saying that, I think all of you involved in the Red Sea Decade expedition knows this beautiful scheme. So the expeditions were in 4 different legs, so covering all the Saudi waters from south to the north, and in different phases. So here we are going to focus in the deepest area, because Al-Sisi was covering the shallower area, and we are going to focus in the work that we did in the Ocean Explorer. The expedition have different goals or themes, and one of them was the biodiversity. And the group, the molecular group in the Carlos Duarte lab, we are focused to develop a comprehensive catalog of the Red Sea biodiversity using metabarcoding and metagenomics tools. So the idea is to analyze the mid-water and coastal ecosystem using sediment and water samples. So I would like also to introduce a little bit the concept of environmental DNA, or eDNA. So all the organisms living in the ecosystem, they constantly lose genetic material to the environment. So just sampling the environment, collecting water, sediment, or even the air, is a very good strategy and non-invasive strategy to to study the biodiversity. So in our case, we collect water samples, and I'm going to explain a little bit the workflow that we did. So we collect water samples. We try to aggregate and condensate all the particles higher than 20 microns and in the filters. So we perform extraction of DNA extraction from the filters, and we try to sequence all these samples. We focus in the CO1 to target the eukaryotic communities. When you have all these sequence, the bioinformatic work starts, and the idea is to produce what we call a MOTU table. A MOTU is an acronym for Molecular Operational Taxonomic Unit that, for the purpose of the talk, we can consider like equivalent or proxy as a species. As Susanna previously mentioned, we were collecting the samples with this bottleneck. That is an oceanographic device attached to an oceanographic rosette. This device was developed by Professor Agustí. I think that for Malaspina, I'm not sure, but I think so. And the advantage of this device is allow you to open and close the bottles in the depth that you are interested. So you can sample all the water column and choose which depth do you want to focus. After collecting the samples, what we did is just to filter the water and store to minus 20 degrees until we perform the DNA extraction. So we divide the ocean in 3 different layers. The epipelagic layer that came from 3 meters to 180, the mesopelagic layer from 180 to 1,000, and the bathypelagic layer that is more than 1,000. And also you can see like what we call here oceanic and coastal, this partially match with we call like phase 1 and phase 2. So in most of the sample station of the phase 2, they were just 2 layers because the bottom depth never arrived to 1,000 meters. And in the right panel, you can see that we deploy 128 stations. And after all the different process, because we lose some samples, we have to remove some samples, some because the bottleneck doesn't close or doesn't open properly, because the DNA extraction were not well, or because we didn't recover enough reads to do proper analysis. At the end, we keep 266 samples covering all the Red Sea. Just some preliminary results, because we are still doing the analysis. I want to tell you that we detected more than 165 million reads. So before to continue the reads, I want you to understand that is every time that a specific sequence has been detected or identified. So it's something that we can imagine like abundance of this sequence. So 25% of the species that we were detected were not possible to assign to a phylum level. But the good point is that this 25% of the species only represent 2.5 of the reads. What does mean? That the species that they were most abundant or most frequently detected, we were able to identify at least to a species level. So to have an idea, we have more than 1,000 species identified at species level, 1,100 species identified at class level, and so on. So at the end, we were able to identify 200 species to a species level. And the idea is improve this resolution with the time. Going a little bit in depth, so the 75% of the reads that we recover from all the water samples belong to the arthropoda, and mainly to copepods. What makes sense because we are talking about the water column and we are talking the zooplankton communities. Just to have an idea, in the literature you can see some traditional studies that in the north of Aqaba, they detected like that the 75% of the total zooplankton community belong to copepods too. So they were able to identify 62 species, with 27 belong to copepods. And on the other hand, another different study for Pradopor in 93 and 19, they recorded that 31 copepod species in the north of Gulf of Aqaba. So the advantage of the new metabarcoding, I want to show you the data that we were able to identify. So we detected more than 1,000 species of arthropods, most of them copepods. It's true that we were not able to give them the name yet, not to all of them, but we can keep this sequence and we know that we have this biodiversity. And in the future, as the databases are improving, we will be able to give it a name to all these species. Also related with the work that Susana did, we found a very high diversity of diatoms. That will be the second group, more than 500 different species. So related with the distribution of the samples, so the legs that was divided the expedition is like an artifact product, so it doesn't have any ecological meaning. So what we have done is, according to Reisus et al., we divide all the Saudi coast in 5 different regions. So it would be the Gulf of Aqaba, the Northern Red Sea, Central North Red Sea, Central South, and South Red Sea. And you can see here the biodiversity that we have found for a very different region. And we have found that the southern part is the most diverse. We have done the same with the different layers, and also you can take a look at the different number of samples, because there is a little bit of bias with the bathypelagic layer, because we don't have too much samples have the other one. The other one are even, and we collect the same number of samples. For the deepest one, we have less samples. But even though the biodiversity is pretty similar, being the mesophytic layer the most diverse one. Just going a little bit deeper, taking account the species richness. The species richness can be measured with different biodiversity index. So the first one will be just the number of species detected, and you have the different regions and the different layers, and you can see there is a smooth trend of higher biodiversity while we are moving to the south. But if we measure a different index, that will be the Shannon Biodiversity Index, we see that the diversity keeps it constant. That means that because this index is not just measuring the diversity, but it's also taking into account the abundance of the species. So we can say that, or we will say that as we are going south, we have most diversity, but most of them is focused in rare species that they are not contribute too much with the abundance. And I love this plot because it can be matched with all that Professor Agustí was mentioned. Here you have the three different layers. So we have the surface, the mesopelagic and bathypelagic layers. And what we have here is how common they are, the species that we have found in the different layers. So and you can see that most of them share, so the most species are shared within the 3 layers, but we have, the 2 layers, the meso and surface, share most of the species. This is related with the sinking particles that Professor Agustí mentioned, because when we sample the water column, we are not focusing the DNA that is just in the environment, but also we are focused in the DNA that is attached to the particles. So since the particles are sinking fast, we were able to detect the similar communities in all the different layers. In this plot, what we can see is again the high abundance. So we are studying the relative abundance of the species diversity, and you can see here again all the regions from the north to the south, and it's coming up the high abundance of the arthropods. So there are some layers, for example, in the central south, that the Chlorophyta phylum is very important, but is a higher dominance of arthropods. And inside this phylum, the most dominant group is the copepods. in this plot, we only represent the phyla with more than 5% of abundance because we were able to detect 49 different phyla in the water samples. Just to finalize, so this is a plot where we have all the samples that we have been collected with a different community structure, and there is a clear pattern that you can see from this side that will be the north regions to the south. And we have represented the main environmental parameters that we have measured around the expedition with the CTD, because we were measuring temperature, salinity, oxygen, beam attenuation, and fluorescence. And what we have detected at the beam attenuation is the main driver of these different regions. The beam attenuation is related with the clarity of the transparency of the water column, and it's related with the primary productivity. So maybe this is related with the phytoplankton bloom that there is occurring more in the southern region than in the north. And just to finalize, I would like to point out the product that we can produce with this kind of a study for NCW. So the idea is to provide a species list with all the samples that we were able to identify, all the species we were able to identify in the water column with our samples. So we can provide a species list. Not all the lists have the species level, but we can check all the taxonomy. And along with that, we also can provide the list of sequence from the Red Sea, that this is something that could be important for the future, because this is something that is not going to change. So as soon as we were able to fill the databases with a better taxonomy, we were able to assign a name to this sequence. And saying that, I would like to thank again to NCW for leading this expedition and giving me the opportunity to talk today. Thank you very much.
Thank you very much, Dr. Carlos. And next to join us will be Dr. Sebastian Schmidt-Roach, a research scientist, marine scientist at KAUST, to talk about the Red Sea Decade Expedition shallow water transit data.
Yeah, thank you so much. I would also like to thank NCW for letting me part on this great expedition, and Professor Carlos for sending me onto a very exciting adventure with the Al-Azizi. So what I'm going to present today is mainly directed towards coral cover and the shallow water habitats that we surveyed by scuba diving and by conducting different transects along the Saudi Arabian Red Sea coast. So this is what has been published in literature as the status of coral reefs of the world for the Red Sea and the Gulf of Annan. And what you can see here is that there has been disturbance, but overall the coral cover in the Red Sea seems to be still relatively okay if you compare it to other places in the world. But this is noticeably based on very limited data, mainly from the northern Red Sea, Egypt and Israel, with very few data points along the Saudi Arabian coast, and also very few data points over longer term, so repeated measuring. So we have an important gap to fill with the expedition based on the Al-Azizi. What we did was a ground truthing exercise where we sent out divers to locations that the Coral Atlas recently published on the distribution of coral reefs and habitats around the world. And you can see here the red is coral reefs. So what we did, we chose very carefully the locations along the coast and sent divers to particular GPS points and to do transects there. You can see all the transects were GPS tracked. You can see how the divers measured about 20 meters along a transect tape, and they had frames, as you can see here, to take photos 1 by 1 meter square frames. So you can see here, sometimes that was very easy because we worked in horizontal habitat, but sometimes it was also quite challenging because the Red Sea has a lot of these walled reefs where the reef just drops down, And we actually had to often be very careful to not drop our equipment because it would have just disappeared into the blue underneath us. But you can see here quite nicely how you have different corals from Acropora, the very pointy corals with the wide tips, to Postulopora that looks a little bit like a raspberry coral, and in between even some encrusting or like massive corals like Poraites. And these is very typical for the Red Sea. But considering that we did a ground truthing exercise, we also had transects that were not very highly covered with corals and that only had a few coral colonies. So if we look into literature, the high levels that were observed in previous exercises are often a little bit biased because people send divers into this very high, very nice, pristine environments, and they ignore a little bit what happens in between. So the coral reef habitats that we monitor is quite representative of what we actually find across the different habitats where coral occur in the Red Sea. So just some facts. If we look from north to south, the species that had the species that had the most range were Poraites lobata, followed by different Goniaster species and an Acropora species. So these were abundant from the far north to the far south. The highest live coral cover observed was near Yambu with 56%. And the mean observed live coral cover, I don't know why it's missing, but it was relatively low, around 2%, but this was based to the fact that we had a lot of habitats where we just had some corals, then substrate, followed by other corals. Generally, the southern reefs around Farasan were characterized by higher cover of non-coral organisms. So here we see the different sampling locations from the north to the south, and these are values extracted from satellite data for the sampling locations. So what I did, I went into publicly available databases, and I could extract for these locations surface temperature maxima in summer, salinity and velocity means currents. So based on this, we can actually extract a lot more information. These are just some examples as a preliminary analysis, and we can go deeply into it and try to see how we can explain certain coral cover patterns, certain species diversity patterns along the gradient. Here you can also see a really nice picture where you can see almost the range that we explored on this expedition from the far south, the Farasan banks, all the way. You can see up there the Al-Wash Lagoon, and then eventually the southern tip of Neom. So if we look at hard coral, live coral cover, we can see that there is a general trend towards the north, having higher cover, but you can see also that at the height of the Farasan banks, the cover comes up, and there's a lot of quite pristine reefs, especially in the offshore regions that we monitored or observed, with high coral cover. The low coral cover in the south at the height of the Farasan can be explained, as Carlos already mentioned, by the recent marine heat waves that we experienced in 2016 and so on. So if we look at some of the reports from, for example, the Living Ocean Foundation, they found in some of the habitats that we also monitored entire stacks of almost 100% Acropora cover, that when we now revisited, we could still see those stacks, but they were eroded and they died off already a time before, but we could see then again recruitment, individual corals coming in again. So we can see it's on a way of recovery, while other places, what is interesting here is quite as well, like if we look at the height of Jeddah, we see that we have some very high, very pristine habitats next to some eroded ones. So all of this will be further analyzed, but it matches already to some degree what has also been reported by the status of the corals of the world, where you can see that the northern Red Sea seems to be relatively stable, while the southern Red Sea seems to be more affected by recent marine heat waves. Notably, you can see the sampling efforts for the southern Red Sea indicated by the colors, so you can see it's often only sporadically monitored. If we look at species diversity, that's quite interesting. we can see also generally a trend to more species diversity based on the Shannon-Weaver index towards the north, but we can see that it doesn't drop off that dramatically towards the south. And that has to do that in the far sun we have a lot of diversity by non-hard coral organism. So for the hard corals, the most abundant genus was Acropora, followed by Porites, followed by Millipora. And that is, if you talk to a coral expert from the Red Sea, not surprising. Those are very common genera that we observe. But now we can look into what is driven and if there are patterns where we find certain genera only in regional, with regional distribution. What we can then also do is we can try to see what drives the species distribution. So here you can see that higher salinity and colder water, it seems to correlate with the abundance of Acropora, meaning that where it's colder, generally Acropora occurs to a higher cover, has higher cover than where it's colder. And you see that, for example, for the fungates, the mushroom corals, they seem to be more found where there is not as much current, while Montipora seems to be thriving in environments with higher current. And so these are patterns that we are currently still exploring to find out more what drives the actual distribution of corals and other organisms, reef organisms, along the Saudi Arabian Red Sea coast. So overall, the transects, and Carlos mentioned this already, did not show any clear signs of coral disease, but we should note that sometimes it's not that easy to spot. But overall, it seemed to be, there were rarely any reports that of a suspected white band or black band disease. And there were only few pale colonies or bleach colonies observed along this whole expedition, but we should also consider that these transects were conducted in the winter and spring season when we don't find that bleach corals are that prevalent. What is quite interesting is that unlike in Australia and the GBR, where crown of thorns starfish is a huge plague, a huge pest, our reefs here seem to be little affected. Although they are there and we could see bite marks and a few transects actually showed their abundance, they don't seem to have a big impact on the health of the corals. So by this I would like to thank again NCW, and we now really are still there on the way to explore this and to dive deeper into this amazing data set. Thank you so much.
Thank you very much, Dr. Sebastian. And now, talking about seagrass blue carbon in the Red Sea, I would like to welcome Hugo Mann, PhD student, marine scientist from KAUST.
Thank you very much, and thank you to NCW for the opportunity to take part in this expedition and for presenting here today. So I'll be talking today about some of the findings from our seagrass survey as part of the Red Sea Decadal expedition, with a particular interest in the blue carbon aspect. So firstly, seagrass is a flowering angiosperm, so it's quite different from a lot of marine vegetation in that it originated in the terrestrial environment and then recolonized the marine environment. And it has these very specific morphological shapes, like its roots, rhizomes and blades, which are not common in, well, don't exist in sort of other marine vegetation, like macroalgae or seaweed. And the rhizomes sort of act to hold the plants together. They connect all of the plants, as you see here. And the blades can be very varied in their size and shapes. You can get very, very large, long, ribbon-shaped blades, and very small, round blades, and anything in between, really. So they're limited in their distribution, but as they are photosynthesizing plants, so they need to be where light's present, and this is limited by depth. and by the presence of nutrients. Yes, so seagrass faces a number of threats. Physically, trawling can tear up the plants, damage them, and tear up the rhizomes, and causes damage to them that way. Pollution, for example, eutrophication or the introduction of too many nutrients to the environment, can lead to an increase in the algae growing on the surface of the plant, and this prevents it from photosynthesizing and causes damage to it that way. And then there are a number of species that not only rely on seagrass, but then seagrass also relies on them. For example, the green turtle forages on the seagrass, and as it clips it, it stimulates growth and increases productivity, as well as increasing the complexity of the habitat, allowing for more biodiversity. And so removing turtles loses this benefit. And then also moving up the food chain, if we remove tiger sharks through overfishing, then this can lead to turtles overgrazing and destroying seagrass meadows this way. So as I say, seagrass supports biodiversity. They are nursery grounds, so due to the sort of habitat complexity and 3 dimensions of it, it offers cover from predation, and so animals can sort of raise their young or leave their young in this area in the hope that they'll be better protected from predation. And there's some economically important species that do this, such as shrimp, and these support coastal economies and food sources. And also seagrass meadows provide grazing for charismatic megafauna such as green turtles and dugongs, and these are sort of big economic draws as tourists like to see these animals. As the seagrass moves in the water, it takes energy from the water column, and the lower energy water column is more likely to drop the suspended particles in it, leading to increased sedimentation in seagrass meadows. And so this builds up a mat, as well as sort of mitigating the energy, and this helps to mitigate onshore wave and storm action. And that increase in the level of the seagrass meadow over time also helps to combat climate change driven sea level rise. And also against climate change, the seagrass meadow sequesters carbon. As I said, it's very limited in its coverage, about 0.2% coverage in the ocean, but it buries about 10% of the carbon, organic carbon buried in the ocean, and is comparable to terrestrial habitats like the more sort of visible and obvious rainforests, but as opposed to these terrestrial habitats where the carbon's stored in the sort of above ground biomass in the plants, in seagrass meadows, the carbon, about at least 90% of the carbon is stored in the sediment. So they store both autochthonous carbon, which is sourced from inside the meadow, so from burial of dead plants, and also autochthonous carbon, which is from an outside source, such as it could be from on land or from algae or mangroves. So as the sedimentation is increased in this place, all the sort of carbon from other sources can come in and be dropped into the seagrass meadow, and it can stay there for up to thousands of years. As I mentioned, there's rhizomes between the plants, create the sort of network that holds the sediment together, and so that prevents resuspension of the carbon in the sediment. But degrading the seagrass through trawling or through decreasing the health of the plant can then lead to the resuspension of the sediment and the reintroduction of all this carbon into the global carbon cycle. So the Red Sea has low nutrients, and there are nutrients introduced from the south at Bab el-Mandeb and minimally through the Suez Canal, and also introduced through dust deposition by the wind. You can see the chlorophyll on the map over there. You're right. And it's much higher as sort of a proxy for productivity. And you can see that that's much higher in the south where the nutrients are introduced, and then there's a gradient moving up towards the north. And you can see this map here shows the number of seagrass species present in different areas of the Red Sea. And you see there's fewer in the north, more in the middle, and then there's again fewer in the south, but this is likely due to sampling bias, as there's fewer studies carried out in sort of the Eritrean and Yemeni waters down there in the south. So to study the carbon and the seagrass community, we took both the photo transects, as Sebastian was just describing, and took cores of sediment to take back to assess the carbon density of the sediment. So the carbon stock that I estimated with seagrass meadows in the Red Sea was 4.89 plus or minus 0.83 kilograms of organic carbon per meter squared. This was extrapolated to a meter depth, which is sort of the standard, and this it allows it to be compared to other environments, but this was developed for terrestrial habitat, so it may not be the best way to do this for seagrass, as the way that the carbon drops off with depth and the sediment isn't the same. But the level of carbon storage is lower than other seagrass globally, but still within the context of Saudi Arabia, it's still a hotspot of carbon burial due to the sort of the arid nature of the environment. So you can see here that I've plotted the carbon density against the latitude, and we have much higher carbon density in the seagrass meadows in the south than in the north, and I've applied a p-value of 0.005 to that, so it's a statistically significant relationship. So yeah, we've got higher carbon storage in the south. And then this is the coverage of different items of the community in different seagrass meadows from the transects. And you can see from the pie charts here, there's some places where there's quite high sand rubble, quite a lot of places where there's quite high sand rubble. And there's also some places where there's only hydroid or macroalgae representing the biota, and so these are places where we've done the transect where seagrass has been indicated, but seagrass hasn't been present at that location. And you can also see that there's places with more monospecificity, so only one species of seagrass present. So there's up at the top there, you can see the Thalassia hempricii and Chamaedosia rotunda dominating. And then there's other places where there's more diverse seagrass meadows. I didn't identify any particular relationship with latitude for that. There's also this scatter plot here showing the percentage coverage of each of these items against the latitude. And as I say, I found no relationship there. So my takeaway is that carbon storage is higher in the south, and sort of underscores the importance of the seagrass as a hotspot of carbon burial for Saudi Arabia. And with this sort of bias, a sampling bias towards the center, I think that it would be beneficial to get more samples, especially for carbon and for the biodiversity in further south. So thank you very much.
Thank you very much, Hugo. And now to give us a presentation about elastomer branches of the Eastern Red Sea, Sofia Frappi, PhD student of Marine Science, KAUST.
Thank you very much for the introduction, and I would like to thank the National Center of Wildlife for inviting me to be part of this symposium. I am Sofia Frappi, PhD candidate in Distinguished Professor Carlos Duarte's lab in KAUST, and today I'm going to talk about the Eastern Red Sea elasmobranchs. So to be all on the same page, the elasmobranchs are cartilaginous fishes, meaning that they possess a cartilaginous skeleton, and they are divided in two huge groups, Bathoidea, that are the rays, skates, and torpedoes, and what we define Selachii or Selachimorpha, that are the sharks. We have more or less 600 species of rays, of Bathoidea, and 500 species of sharks in our planet. And they have a very long evolutionary history that date back to the Devonian, more than 400 million years ago. So they are very ancient creatures. And they went through 3 mass extinction events, and they survived. So they are very resilient. But nowadays they have to face the Anthropocene era with a lot of threats. For instance, the unregulated and intensive fisheries, the climate change and habitat degradation, and everything is exacerbated by their life history traits. For instance, they have a very slow rate of maturity and growth and low reproduction rate. So all of this makes them even more prone to extinction. In some area of the world, in fact, in the last decade, we lose more than 90% of the elasmobranchs population. For instance, this happened in the Mediterranean and in the Northern Atlantic. In 2008, in the Kingdom, there was a ban on any kind of shark fisheries-related activity. But as always, it's very hard to enforce this kind of policy for protection. And in fact, we still need to conserve and restore those populations. And this, it needs to be done timely. But to know what we want to restore, we need to have a broad overview on the ecology and habitat use of this population. And in these studies, in fact, we aim to investigate the ecology, the habitat use, the depth extension range, and the current status of this population in the Red Sea. So we gathered the observation during the Red Sea Decade expeditions using the 2 vessels that were available, so Al-Azizi and Ocean Explorer, and the observation were gathered through different methodology, the ROV, the 2 submersibles, the helicopter surveys, and the scuba diving surveys. Moreover, we were also able to collect abiotic factors like temperature, salinity, oxygen, and so on, every time we were seeing a shark or a ray. So this is the very first publication that came out using the dataset of observation from the Red Sea Decade expedition. And this publication is about an extension in the depth range at a global level for different species of elasmobranchs. In particular, the Carcharinus altimus, the big-nosed shark. We were able to see it during an ROV dive at 886 meters, and it has never been recorded so deep anywhere else in our planet. In fact, the maximum depth recorded was 810 meters before this expedition. And this is a very peculiar fact because this is a predator with a very high metabolism, and in other areas of the world it would not survive so deep because of the temperature, as Professor Carlos was explaining before. So here, the peculiar temperature of the Red Sea that stays high, even going very deep, allow this kind of animals to survive and thrive even in deeper water. Here, the first record of feeding behavior of the little Iagomanensis, the big high hound shark, that is a typical shark of the deep in the Red Sea. In fact, we recorded a lot of sightings for this species. So this data set has been used also to prepare another publication that is going to be about the distribution and ecological interaction of the Eastern Red Sea elasmobranchs. And we use the data set coming from the Red Sea Decade expedition together with some data that we extracted from literature from previous studies in the Red Sea. In the map, you can see that the gray dots are the data coming from this expedition. And in fact, the survey effort is huge as we gathered observation along the whole coast of Saudi Arabia. And Going a little bit more into detail, you can see in the pie chart the number of observation for Batoidea and sharks that we gathered during the expedition. And on the bar plot on the further right of the screen, you can see these sightings divided by different methodology through which they were gathered. So what you can see, and is very striking, is that helicopter surveys were very good in gathering information about Batoidea. And this huge value is also thanks to the fact that we found a huge group of mobule in the southern part of the Red Sea through an helicopter survey. And you can also see that drop cameras, ROV, and submarine dives were the best method to gather information about sharks. So, as I said before, we also were able to gather abiotic information, so temperature, oxygen, salinity, for instance. Here you can see represented the temperature and oxygen, thanks to the fact that we had a small CTD mounted on the ROV. So every time we saw a shark, we would take the temperature at that depth or the oxygen value at that depth. And then we used them, we plotted down, and here you can see the fundamental requirement in terms of oxygen and temperature for different genera of elasmobranchs. And another thing that is very important is that some of these ellipses contain other ellipses, meaning that with one genera of sharks can encompass also the fundamental requirement for other genera and other species. And this aligns with the concept of umbrella species, for which if we protect one species, we can potentially protect many more species. And this is very important when it comes to conservation efforts. So this is a very important analysis that we run, that is the sighting per unit effort analysis. So we decided to use all the information gathered through the Red Sea Decade expedition, and then we also went through literature, and we used the data coming from literature of previous published papers on the Red Sea. And we compare the data from this analysis in the Red Sea with the same kind of analysis coming from other areas of the world. And what we see that both for deep ocean, for the deep sea, and for the shallow coral reef, the values for the Red Sea are very, very low. And what does it mean? It means that, unfortunately, in the Red Sea, we can see less sharks and less rays in respect with other areas of the world. And this can be translated into a pattern of declining populations of elasmobranchs in the Red Sea. And this calls for further effort in their protection and maybe enforcement of the policy for their protection. So next, I have some highlights of an important observation during the Red Sea Decade expedition, and I hope you will enjoy. National Center of Wildlife for giving me the opportunity to be part of this expedition, and my professor Carlos Duarte for giving me the opportunity to work on this amazing project, and to all the crew of the Al-Azizi and the Ocean X that helped us through the entire expedition. Thank you.
Thank you very much, Sophia, for that very amazing and interesting presentation. Next, I would like to ask Megan Nolan, PhD student and marine scientist, to talk about biodiversity and distribution of mesophotic and deep coral reefs in the Red Sea.
Hello, thank you for the opportunity to be here, and thank you everyone for staying to the end of the day. My name's Megan Nolan. I'm a PhD student at KAUST in Professor Francesca Benzoni's Habitat and Benthic Biodiversity Lab, and I'm going to be presenting today on some of the projects that I've been working on with data from the Red Sea Decade Expedition. This comes from my PhD thesis titled Biodiversity and Distribution of Mesophotic and Deep Coral Reefs in the Red Sea. So first, I'd like to take a step back and look at vertical zonation on coral reefs. So we're all quite familiar with shallow coral reefs, similar to the one you see in the picture here. These are found between down to 30 meters depth and are pretty well known and very well studied. Below this, we have mesophotic coral reefs found between 30 and about 200 meters depth. These have been much less studied due to the difficulties in accessing them and as they fall below the depths of recreational scuba diving. And even deeper than this, we have deep reefs. These are found below 200 meters depth where there's no longer any light available to support life forms. And they're very remote, very difficult to access, and therefore we know much less about them. So this represents a significant gap in our knowledge. So you can also see differences in the different depth zones when we look at the oceanographic conditions of the Red Sea. So this figure on the right shows fairly consistent trends going from the northern Red Sea on the left down to the southern Red Sea on the right, where we have, and when we look at the different depth zones, we can see that in shallow and mesophotic waters, we have a lot of variation in these environmental conditions, whereas in the deep, these are very stable conditions. All of these oceanographic parameters make the Red Sea a very interesting and unique area to study, and also mean that we have the potential to have very unique associations and species in our deep waters here. So these are the data sets that I'm going to be focusing on from the Red Sea Decade Expedition. Firstly, the bathymetric data, which we've processed to be used in future projects. Then we have the video transects from the submersibles and the ROV. And finally, the environmental data that was collected at CTD casts at point locations. And with this data, I wanted to understand the spatial distribution of coral-dominated associations in the mesophotic and deep Red Sea. So I separated this into two questions. The first is how can we characterize the coral-dominated associations that are in the mesophotic and deep Red Sea? And we approached this using quantitative baseline surveys with the video transects from the ROV and subs. The second question was about how can we model the distribution of important benthic organisms in underexplored areas, and we hope that this will result in predictions across the whole of the Red Sea. So starting with the image analysis, this is the distribution of the data we had with 125 ROV dives and 20 submersible dives, and then we wanted to characterize the association at each point in the video. So by association, I mean a group of organisms co-occurring in time and space, where we have a key structuring organism like the coral, and then associated fauna, including crustaceans, sponges, and fish, for example. So in order to classify the association, we looked at the dominant groups, which were the structuring organisms, any secondary groups, the substrate type, whether it was sandy or rocky, for example, and the depth range. And then we wanted to quantify this, so we used Reef Cloud, which is a point-based analysis that uses AI integration on extracted images. So this is how it works. Here's an example image, and it has 50 points randomly overlaid over it. So I extracted 12,000 images in total, resulting in 600,000 points. I manually annotated 15% of these, and after that 15%, the model was sufficiently trained to be able to start to recognize these categories by itself. So for example, it could identify these two corals as gorgonians and could identify this point as being on top of rock. So I have some results from the initial video analysis. we identified 12 distinct coral associations. So coral was present in all 12 of these, but it was the dominant structuring organism in 8 of them. So I've included some images here, and you can see that coral is present, whether it's stony corals, black corals, or gorgonians, is present in all of these. We also identified one completely new association to the Red Sea, and that's the sponge grounds, which I'll talk a bit more about in a second. So this work has already been presented at Asia Pacific Coral Reef Symposium last June in Singapore, and I'm currently preparing a manuscript. So I wanted to show some of the distribution from some example associations. So this first one is for a Caryophyllae framework. So this is a kind of deep sea reef built by a specific coral family. And you can see here we found it fairly frequently along the coastline from the northern Gulf of Aqaba down to the southern Red Sea. My second example is a Dendrophilidae framework, so another type of deep sea reef built by a different family of corals. And this is actually the same type as was first identified by Caban et al. In 2020 in the northern Red Sea. So we found these in slightly shallower water between 250 and 500 meters, but with a similar spatial extent. And finally, the sponge grounds we found in 4 locations associated with the blue holes in the southern Red Sea. These are built by dense aggregations of the sponge Triticalex poli, which is a sponge species that we only have in the deep Red Sea. It isn't found anywhere else in the world. They're also associated with Stylasta tritoni, a lace coral which is also only found in the Red Sea. I also want to draw your attention to the brown areas that we see between these white organisms, and this is dead sponge matter. So you can see also from this graph that this dead sponge takes up a large part of the area, and that just shows that it's very well retained within the habitat and can provide important resources for other species which may depend on it. So as well as looking at the associations as a whole, we also wanted to look at the species which form them and understand more about where they are distributed. So we identified locations of species from the videos. For example, this species of stony coral, Madrassus interjecta, was found at all of these points, was observed at all of these points from the video transects. So we targeted key association-forming species, as I mentioned, and made sure they were common and easily identifiable from the videos. So these were the other 4 species that we selected. That's Stylasta tritoni, which is a lace coral, the black coral, Bathypathes thermophila, the other stony coral, Dendrophilia sp, and finally the genus of octocoral, Acanthogorgia. So before we can understand where these species are distributed, we first need to understand what controls their distribution, or what are the drivers. So these drivers can be biological, as we see on the left, geological or geomorphological, oceanographic or anthropogenic. A lot of these are very hard to study and cannot be measured quantitatively, but we do have some examples, and for example, these 6 measures, which have already been processed from OceanX, from the Red Sea Decade expedition. So these were the ones that we used in our models. So we first started with the with the bathymetric data, which I mentioned earlier has already been processed. And then we combined that with other bathymetric data from different sources in order to have complete coverage across the Red Sea. We were then able to extract different descriptors of the seafloor, including, for example, the steepness of the slope or the aspect, which is the direction of the slope. And then we wanted to combine this with environmental parameters, including temperature, salinity, and oxygen concentration. And Alex mentioned how these parameters were measured earlier. So we used the CTD data, and thanks to a collaboration with Professor Duarte and Professor Assis in Portugal, we were able to then model these values using an existing pipeline called BioOracle. And that will result in maps similar to the one on the left, on your right, which will cover the whole Red Sea at the seafloor. So then when we create these models, we first start with the presence locations, as you see here for Madrassus interjecta. We combine this with seafloor descriptors, which are taken from bathymetry, and the environmental data, which comes from the CTDs and the models. And finally, this will result in a predicted distribution across the whole Red Sea for our species of interest. So this is example data, but this is the output that we hope to get. So this, in red, shows the potential distribution of Madrassus interjecta. And these results will provide the potential distribution of important organisms across the Red Sea. It will allow us to study them from latitudinal depth and cross-shelf gradients, and as well as their ecology, we can also use this to target further exploration and identify areas such as this one here, the Gorgonian Association, and try and figure out where they may be found. So it's really important to study the associations that we see in the deep Red Sea, as they've been known to attract other species, including anemones, shrimp, squat lobsters and fish, which have all been seen to live in coral associations in the deep. They also represent a potentially large and currently unknown light-independent carbonate factory in the deep Red Sea, and they may have an influence on marine biogeochemical cycles at potentially globally relevant levels, particularly the deep coral frameworks. This also could be an important consideration in blue carbon calculations in the region. And finally, understanding the distribution of these associations and species is incredibly important for conservation. And this is what I plan to do next, particularly in light of Vision 2030 and the ongoing Giga projects in the region. I hope to conduct conservation analysis with the results of these models. Thank you very much to Professor Duarte, Dr. Caban and Dr. Peribone for all of the organisation before, during and after the Ocean X expedition, and thank you for listening.
Thank you very much. And with that, we conclude our sessions for today and hope you guys had a very enjoyable and meaningful symposium today. Tomorrow we will continue with our talks and hope to see you guys then. And for now, have a good day.